Start na po. This is microbiology and parasitology ratio. Kamusta guys? Hello? Hello po? Narinig niyo ako?
Yes, sir.
Sige, start na din tayo. I just want you to know that some of the items are the items that I wish I knew before I took the boards. Kaya sinishare ko din sa inyo. Stop. Okay. Number 1. A non-living entity that is contaminated with etiologic agent and as such is the mode of transmission for that agent. What's the answer? A, agents, B, vector, C, vehicles, vehicle, D, reservoir. When we say agent, this is the actual agent that caused the disease or the causative agent. Vector is the entity that is contaminated with etiologic agent and also act as a mode of transmission, but it's a living entity. The correct answer here is vehicle. A non-living entity that is contaminated with etiologic agent and as such is the mode of transmission. Example, example, water, the soil, and other inanimate objects. When we say reservoir are the source of the agents that provides the pathogen with an adequate condition for their survival. Number 2, he concluded that cholera, again guys, a non-living entity that is contaminated with etiologic agent and as such is the mode of transmission is vehicle. Kapag living entity like insects, they are vectors. Number 2, he concluded that the cholera was spread via fecally contaminated water. The water at the Broad Street pump was being contaminated with sewage from the adjacent houses. He is considered by many to be the father of epidemiology. A, Joseph Lister, B, Paul Ehrlich, C, John Snow, D, Anton Leeuwenhoek. Si Joseph Lister considered as the father of antiseptic surgery. Si Paul Ehrlich naman, kilala siya sa microbiology as the Mr. Salvarsan 606. It is a arsenic compound that acts against syphilis. Sa immunology naman, kilala siya as sa phagocytosis, pero mas kilala si Elie Metchnikow. And he also coined the word autotoxicus. The body reacts to self-antigen. Si Paul Ehrlich din yun. Now, si John Snow, letter C is the correct answer, father of epidemiology, John Snow. Si Anton van Leeuwenhoek naman is the father of microbiology and a father of what? A father of protozoology. Pero pag tinanong who is the father of a modern microbiology, hindi si Anton yun. It's Louis Pasteur. Louis Pasteur is the father of modern microbiology. The father of microbiology and protozoology is Anton Leeuwenhoek. The correct answer here is John Snow. Pero kapag tinanong naman who is the father of hospital epidemiology or a father of hand washing, a saver of mothers, iba naman yun, si Ignace Hemingway. he discovered the importance of hand washing. Nung na-discover niya yun, bumaba yung maternal death sa isang maternal hospital. And he is the father of hand washing, Ignace Hemingway. Ito, what is the purpose of adding 0.025% of sodium polyinyl sulfonate to nutrient-broth media for the collection of blood cultures? A, it inhibits phagocytosis and complement. B, it enhances growth of anaerobes. C, it functions as preservative. D, it promotes blood clot. C, D, it promotes blood clot, mali. Kasi SPS or polyinyl sulfonate act as an anticoagulant. So it doesn't promote formation of a blood clot. And sa C naman, it functions as preservative, hindi. Well, in fact, inihinhibit niya yung activity ni aminoglycosides. And letter B, it enhances growth of anaerobes, mali din. Why it inhibits the growth of other anaerobes and Neisseria gonorrhoeae. The correct answer here is letter E. It inhibits phagocytosis and complement. Ano na nga yung it neutralizes the effect of SPS? It's 2% gelatin. It neutralizes the effect of polyinyl sulfonate. It's 2% gelatin. Ito. Cerebrospinal fluid should be cultured immediately, but if delayed, the specimen should be A, refrigerated at 4 degrees to 6 degrees. B, frozen at -120 degrees. C, incubated at 37 degrees and cultured as soon as possible. D, stored at room temperature for no longer than 24 hours. The correct answer here is C, incubated at 37 degrees and cultured as soon as possible, as long as that CSF is not it's not a viral specimen. Kung viral specimen, then you have to refrigerate it at 4 degrees Celsius. Ito, paki-memorize ako, table na to. Kapag 4 degrees Celsius, you have to store urine, stool, viral specimen, except for genitals, foreign devices like catheters, stool for C. difficile toxin, study up to stool for C. difficile toxin, study up to 3 days. -20 naman degree kapag sa room for serologic studies that may be frozen for up to 1 week. -70 tissues or specimen for a long-term storage, including stool for C. difficile toxin study if testing is delayed for more than 3 days. CSF should always be at 37 degrees unless viral specimen siya, and it should be stored at 4 degrees. 22 to 25 room temperature. This is for anaerobic culture, sterile body fluids, and genital specimen swabs. Number 5, a gram-stained sputum smear which shows 40 to 50 squamous epithelial cells per low power field, along with gram-positive cocci, many gram-negative rods, and many gram-positive cocci in pairs using oil immersion objective. How should the laboratorian or a medical technologist report the result of this smear? How? A, call physician directly to report a life-threatening situation. B, subculture must be done to confirm, then the report is pending. C, report gram-positive cocci, many gram-negative rods, and many gram-positive cocci in pairs, as well as many squamous epithelial cells. D, no reports and request another specimen. The correct answer here is letter D. Meron tayong tinatawag na Bartlett's criteria that if the sputum does not don't meet this criteria, then you have to reject it. Ano yun? Negative, it should be lesser than 10 dito. It should be lesser than 10 epithelial cells per low power field and greater than 25 polymorphonuclear cells. Si Bartlett classification, it used to assess the quality of sputum samples. It is recommended that all specimens containing purulent secretions should be cultured, whereas those containing oral secretions be recollected. Paano natin malalaman kung oral secretions lang or pure saliva, we can grade it, and we grade it with the number of neutrophils per LIF, per low power field, and number of epithelial cells per low power field. Kapag fewer than 10, then the score is 0. Kapag 10 to 25, the score is plus 1. Kapag greater than 25 neutrophil, the score is plus 2. Pag mucus lang, plus 1. Ito, kapag yung number of epithelial cells naman, kapag 0 to 10 ang epithelial cells or squamous epithelial cell per LPF, then we have to deduct negative 1, minus 1. And kapag greater than 25, minus 2. And take note that the total scores of 0 or less indicate lack of inflammation or presence of saliva. Let's say, for example, kapag nakita ka ng 15 number of a neutrophil, which is 15 per low power field, anong score niya? Plus 1. Pero nakakita ka ng epithelial cells of 15 din epithelial cells per LPF, minus 1. Total score niya is 0. So reject this specimen kasi saliva lang siya. Pero ang importante ito, tandaan nyo, the Bartlett's criteria. Kapag lesser than 10 epithelial cells per LPF, then greater than 25 MNS polymorphonuclear cells, in order for you to process the sputum. If not, then reject it. 6, laboratory professionals are at risk for disease transmission. The majority of cases of laboratory-related infections are associated with A, infectious aerosol, B, puncture wounds, C, contaminated of abraded skin, and D, person-to-person transmission. The correct answer here is letter A, infectious aerosols. They are airborne kasi aerosols may form during centrifugation of unstoppered tubes or form heating cultures or specimen too rapidly. Then, sabi pa dito, leakage from container that holds contaminated specimen. So the majority of cases of laboratory-related infections are infectious aerosols. Number 7, legally responsible for ensuring that an exposure control plan has been implemented and not mandated, and that mandated safety guidelines are followed. Legally responsible for ensuring that an exposure control plan has been implemented. A, medical technologists and medical technicians. B, medtech and pathologists. C, laboratory director and supervisor. D, microbiology supervisor and safety officer. The correct answer here is, in this question, is C, laboratory directive and supervisor. Nagkamali ata ako ng pag-bold nun sa answer key. But the answer here is laboratory director and supervisor. Kapag post-exposure naman, microbiology supervisor and safety officers. Kapag post-exposure. Kapag exposure control plan, ulitin ko, laboratory director and supervisor. Now, importante din to, number 8. Diluted household bleach or sodium hypochlorite inactivates HIV in blank and HBV or hepatitis B virus in blank. The answer here is 2 minutes sa HIV, then 10 minutes sa Hepa B virus. Again, diluted household bleach inactivates HIV in 2 minutes and hepatitis B virus in 10 minutes. Okay, number 9, to eliminate the antibacterial properties of blood and simultaneously introduce an adequate volume of blood for recovery of microorganisms from septicemia, the recommended blood to broth ratio in the blood culture battle is approximately A, 1:2, B, 2:1, C, 1:10, D, 10:1. Again, recommended blood to broth ratio. Ang sagot is 1:10. Kapag broth to blood ratio pinagbaliktad, 10:1. Okay, which of the following organism is milk-borne disease for humans? A, M. tuberculosis, B, Bacillus anthracis, C. S. typhi, D. Q fever. Now, kapag milk-borne disease, these are disease that is transmitted via milk. And when we say milk-borne disease for humans, these are milk-borne disease that came from a bovine origin. Kapag tinatawag din natin milk-borne disease for cows or bovine, this milk-borne disease came from human origin. Letter B. Actually, letter B and letter D. Kung sinagot yun yung D, correct din. B anthracis and Q fever. Ito, pa-memorize ako. Sabi dito, milk-borne disease transmitted through milk may be the result of either disease cow or bovine origin or disease human handlers or human origin. Kapag bovine origin, it is a milk-borne disease for humans. Kapag human origin, milk-borne disease for bovine or for cows. Under bovine origin, number 1, bovine tuberculosis. Huwag malilito kasi sa human origin, andito si M. tuberculosis, human tuberculosis. Antulant fever by Brucella abortus. Then anthrax, Bacillus anthracis, cowpox virus, foot and mouth disease, aptovirus to, foot and mouth disease. And 6 is Q fever. Human origin naman, typhoid fever, salmonella, scarlet fever, cholera, diphtheria, septic sore caused by group A streptococcus, and human tuberculosis. Now, which of the following filters can give 10% sterility? A, Sheets, B, millipore, C, millipore 0.22 micrometer, C, millipore 0.5 micrometer, D, both A and B. Correct answer here is letter B. Now, colistin nalidixic acid, agar, is used primarily for the recovery of A. Neisseria species, B. Enterobacteriaceae, C. Pseudomonas aeruginosa, D. Staphylococcus aureus. The correct answer is letter D, Staphylococcus aureus. Now, chocolate agar and modified Thayer-Martin agar are used for recovery of what? A. Haemophilus species and Neisseria species, B. Haemophilus species and N. gonorrhoeae, C. Neisseria species and Streptococcus species, and D. Streptococcus species and Staphylococcus species. The correct answer is letter A. Correct din sana yung Haemophilus species and N. gonorrhoeae. Kaso, hindi lang si N. gonorrhoeae, other Neisseria species can thrive in what? Used in the recovery of they are recovered in chocolate agar. And si modified Thayer-Martin is enriched chocolate agar siya, enriched chocolate agar or modified Thayer-Martin. So nagtathrive din si Haemophilus at ang Neisseria species. Ngayon, si Neisseria gonorrhoeae, hindi siya nagtathrive or cannot be recovered in BAP. Cannot be recovered in BAP or blood agar plate. Chocolate agar lang siya. And Thayer-Martin. In contrast kay Moraxella catarrhalis, nag-grow siya sa cup and BAP, including Neisseria meningitis and meningitidis and Neisseria elongata. Ito. In search for carriers of Salmonella typhi, the plating medium of choice is what? Ano sagot niyo dito, guys? Letter B. Yes, correct. Huwag niyo pabalik na rin Salmonella shigella, hindi Shigella salmonella. Salmonella shigella, selective for Salmonella and Shigella species. Kapag bismuth sulfite naman, highly selective for Salmonella typhi, where it has black colony surrounded by black zone with metallic sheen. The other Salmonella, they exhibit green colonies. Si brilliant green naman, na-inhibit si S. typhi and si Shigella. S-typhi and Shigella are inhibited in brilliant green, but they are si brilliant green highly selective for Salmonella other than S-typhi. Si Eosin methylene blue naman, isolation and differentiation of lactose fermenting and non-lactose fermenting enteric bacilli. Now, which of the following organisms would be negative using acid fasting? Ano sagot niyo dito? A. Cryptosporidium, B. Azospora belli, C. Actinomyces israeli, D. Nocardia asteroides. What's your answer? Ano sagot niyo? Po? Letter C. Yes, correct. Pamemorize ako kasi importante ito. Modified acid fast. Isospora, Cyclospora, Cryptosporidium, Nocardia, Cordonia, Rhodococcus, Sucamorella, and Legionella myctidae. These are acid fasting. Si Actinomyces israeli is not an acid fast. Now, number 16. Before reporting a negative AFB smear, the smear must be examined carefully by scanning at least blank oil immersion fields. Ano sagot?
300, sir.
300, correct. Before ka mag-report ng negative, then make sure that you scan 300 fields per oil immersion fields. Ito, how will you measure the zone of growth inhibition for the Gerby-Bauer susceptibilities? How? A, measure it on the underside of the plate. 2, using a ruler, caliper, or template. 3, with an aided eye. A, 1, 2, 3, B, 1, 3, C, 1, all except 3. The answer here is letter A, 1, 2, and 3. Wala sigurong nagka-count under microscope. The correct answer here is all, 1, 2, and 3. Now, performing oxidase tests, which of the following techniques can be used? A, rub colony on a filter paper strip and add a drop of the reagent. 2, put a drop of reagent on the colony. 3, rub the colony on a piece of filter paper containing the reagent. A, 1 and 2, B, 1 and 3, C, 2, and 3, D, all of the above. All are correct. The correct answer here is all are correct. Letter D. Yun. Similar at 2, 1A, but has 100 IFM intake air velocity and plenums are under negative pressure to room. Exhaust air can be ducted to the outside through a canopy unit. A class 1, B class 2A2, C class 2, class B class 2A1, C class 2A2, D class 2B2. The correct answer here is class 2A2. Huwag kayong malilito kasi class 2 B3 is class 2A2. Same lang sila ni class 2A2. Either tawagin mo silang class 2A2 or class 2B3. Now, si class 1, it allows room air to pass into the cabinet and around the area and material within, sterilizing only the air to be exhausted. Now, sa si class 2A1 naman, take note, 70% air-recirculated and 30% exhaust. Huwag pagpalit na rin sa class 2B2 ay class 2B1. 30% is recirculated and 70% is exhaust sa class 2B1. Si class 2B2 naman, air discharge outside the building. Gumagamit tayo ng class 2 biosafety, class B biosafety cabinet if it involves what? Radioisotopes, toxic chemicals, or carcinogenic chemicals or substances. Correct answer here is letter C. Okay. A urine specimen was collected at 7 a.m. The nurse labeled the sample and placed it next to the tube station to be sent to the laboratory for testing. The specimen was received in the laboratory for culture at 1 p.m. on the same day. The microbiologist or a technologist should what? A, check for the possible contamination and perform the test. B, perform only gram staining. C, set up a routine bacteriology culture and note the time. And D, notify the nurse why the specimen is rejected and request a new sample. Ano sagot niyo dito? Anyone? What's your answer here? Correct answer? B, yes. Notify the nurse. Why? Kasi sabi dito, a preserved urine should be processed within 24 hours. Preserve. Pero dito kasi hindi siya na-preserve. It's actually unpreserved. Kapag unpreserved urine, you have to process it within 2 hours or less than 2 hours at room temperature. Now, when multiple specimens arrive, at the same time, priority should be given to those that are most critical. Priority should be given to those that are most critical. Examples of these specimens are what? ACSF, tissue, blood, and sterile fluid. B, urine, throat, sputa, stool, or wound drainage. C, acid fast, viral, and fungal specimen. D, all of the above. What's the answer?
A.
A, correct. Tandaan yung CTBS. CSF, tissue, blood, and sterile fluid. And take note that the acid fast, viral, and fungal specimens, they can be processed by batch. Per batch nyo lang siya ipaprocess. Again, CSF, tissue, blood, and sterile fluid. This should be given priority. Number 22, all of the following are appropriate when attempting to isolate gonorrhea for a genital specimen except A, plate this specimen on a modified Taylor Martin. Plate this specimen on a New York City agar. C, culture specimen in ambient air at 37 degree. D, transport the genital swab in charcoal transport medium. Sorry. Correct answer here is letter C. Hindi, wag mo siyang i-culture ambient temperature kasi these are for aerobes. And si gonorrhea capnophilic siya. Kapag aerobes, ambient air which contains what? 21% oxygen and small amount of syrup of carbon dioxide. Ang requirements kay gonorrhea, it requires 5 to 10% carbon dioxide and approximately 15% oxygen. Capnophilic. Capnophile. Now, what is the number of bacteria needed to cause turbidity in a broad culture and be seen with unaided eyes? Na kapag if the bacteria reaches this certain number, nagmamanifest siya sa broth as as a turbid solution. Anong sagot niyo?
C.
C, correct. C, paano ang ganito tanong? Kapag nakita ka ng isang bacteria per OIF, approximately how many organism per ml of the broth of the substance? Approximately how many organism per ml? Kapag nakita ka daw ng bacteria per OIL, approximately how many organism per ml? It's 5 is to 10. 10 raised to 5 or 100,000 organism per ml. Ito, 10 is to 6. The number of bacteria needed to cause turbidity in broth culture and to be seen with unaided eyes. 10 is to 6. Kapag isang bacteria per OIO is equivalent to 10 is to 5 organism per ml. Ito, apple green fluorescence with FITC dye, A1+, B2+, 3, C3+, D4+. Ano sagot nyo?
2 plus. B.
Apple green. Kapag negative, no apple green fluorescence. Kapag 1+, faint yet unequivocal apple green fluorescence. Pag 2+ naman, apple green fluorescence. Kapag 3+, bright apple green. Kapag 4+, brilliant apple green fluorescence. Ito, susceptibility testing performed on quality control organism using a new media lot number yielded zone sizes that were too small for all antibiotics tested. The testing was repeated using a media from a previously used lot number and all zone sizes were acceptable. Which of the following best explains the unacceptable zone sizes? Ngayon daw, kumuha ka daw ng bagong media and then na-control ka, they yielded smaller zone. Pero nung kinuha mo yung dating media na ginagamit mo, they were all normal. So what's the reason behind? A, the depth of a media was too thick. B, the depth of the media was too thin. C, the antibiotic discs were not stored with a proper desiccant. D, the antibiotic discs were not properly applied to the media. Ano sagot nyo?
B.
Ano?
B.
The depth of media was too thin. Remember, it was too thin. So, 4 millimeter lang siya, lesser than 4 millimeter. Kapag too thin, then it should yield a larger zone. The correct answer here is letter A. The depth of a media was too thick. Ito ibigay ko sa inyo yung mga problem niya and then yung effect niya, pa-memorize na lang. Kapag ang Mueller-Hinton medium is too deep or greater than 4 millimeter, then the zone sizes will be too small. Same dun sa tinatanong kayo na sa question, the zone sizes will be too small. Kapag yung Mueller-Hinton medium is too shallow or lesser than 4 millimeter, then the zone sizes will be too large. Take note, will be too large. Kapag yung inoculum naman, the inoculum is too heavy, then the zone sizes will be too small. Kapag yung inoculum naman is too light, zone sizes will be too large. Huwag pagbalik na yun. Zone sizes will be too large. Tapos yung pH ng Mueller-Hinton agar, too high or too low, ano yung effect niya? May affect the activity of some antimicrobial agents. Kapag yung this that is placed too close together, effect niya, may affect the activity of some antimicrobial agents. The this nut press flush with agar surface may result in irregular ship zones. Kapag yung inoculated plates incubated in carbon dioxide incubator, the carbon dioxide could diffuse into the medium, causing the formation of carbonic acid, which would lower the pH of the medium. Kapag nag-low yung pH ng medium, may affect the activity of some antimicrobial agents. Ito, the two cations known to influence the activity of aminoglycosides. A, sodium and potassium. B, calcium and magnesium. C, calcium and potassium. D, sodium and magnesium. Ano sagot niyo dito?
B.
Yes, calcium and magnesium. At what concentration? Calcium 25 mg per deciliter. Yung magnesium naman 12.5 mg per deciliter. Kapag increase yung dalawang cations na to, then expect that there is a decreased activity of aminoglycosides to P. aeruginosa, and there is a decreased activity of tetracycline against all organisms. Again, decreased activity of aminoglycosides to P. aeruginosa and a decreased activity of tetracycline against all organisms. Kapag thymidine naman, kung ang nag-increase, when there is increased thymidine content, it causes false resistance to sulfonamides and trimethoprim. It causes false resistance to sulfonamides and trimethoprim. Kapag low pH, decreased activity of what? Of aminoglycosides erythromycin, clindamycin, and increased activity of tetracycline. Again, pag low pH, decreased activity of aminoglycosides, erythromycin, clindamycin, and increased activity of tetracycline. High pH naman, decreased activity of tetracycline. Pa-memorize ako nito. Ito. It is considered as an antimicrobial agent but not as antibiotic because it is not produced by microorganism. A chloramphenicol, B tetracycline, C erythromycin, D sulfonamide. Anong sagot nyo?
C.
Is it erythromycin? It is considered as antimicrobial agent.
Sulfo naman.
Letter D, sulfonamides. They are not produced by organism. Pero yung tatlong antibiotic na ko, they came from an organism. Ito, antimicrobial susceptibility testing occasionally required for this organism. Occasionally required, AST. What's your answer? A Acinetobacter. B Enterobacteriaceae. C Enterococci. Moraxella catarrhalis. Yes, correct, Moraxella. Ito ha, this table, this is the categorization of bacteria according to the need of routine performance of antimicrobial susceptibility. Kapag commonly required, again, kapag commonly required, ito, Staphylococci, Streptococcus pneumoniae, Viridans streptococci, Enterococci, Enterobacteriaceae, Pseudomonas aeruginosa, and Acinetobacter species. These organisms are commonly required in AST. Kapag testing occasionally required naman, si Haemophilus influenzae, Neisseria gonorrhoeae, Moraxella catarrhalis, and anaerobic bacteria. Kapag really required naman, really lang siyang nire-required, the beta hemolytic streptococci, the Neisseria meningitidis, and the Listeria monocytogenes. These are really required for AST. Pa-memorize na lang. Ito. Which of the following has a mushroom-like odor? Lumabas to sa amin noon. A. Pasteurella multocida. B. Pseudomonas aeruginosa. C. Proteus. D. alkalinogens. Alkaline. Letter A. Kapag burnt chocolate cake naman, si Proteus yun. Kapag mushroom-like odor, P. multocida. Kapag freshly plowed grass, nocardia, old socks, S. aureus, peach naman, echinella, or H. influenzae. Now, a spherical gram-positive cocci that is catalyst positive, non-motile, and unable to produce acid from glucose anaerobically is best identified as a genus of what? Letter A. Letter A. Ito. Coagulase positive, PYR negative, acetoin positive, or Voges-Proskauer positive, Staphylococcus. Ano sagot nyo? Staph. Yes. Letter A. Ito. Staph positive sila. Si Lignonensis positive din siya as coagulase. Coagulase positive din siya. Hindi ko lang na-include dito. Pero ang tatandaan nyo sa kanya, oxacillin resistant si Lignonensis. Ngayon, these are the tube coagulase. S. aureus, intermedius, take note variable sila, si coagulans and hyacus. Kapag Voges-Proskauer, positive si coagulans and si aureus. Kapag PYR naman, positive tong dalawang to. Si S. intermedius and coagulans. Si staph ba, bacitracin susceptible ba siya or resistant? Si aureus ba? Si sao? Bacitracin resistant ba siya or susceptible?
Resistant.
Resistant. Si strep. Strep pyogenes. Is it resistant or susceptible? Susceptible. Okay. Resistance to clindamycin can be induced in vitro by A. Ampicillin, B. Erythromycin, G. Gentamicin, D. Penicillin.
Gentamicin.
Meron tayong tinatawag na ganun, na inducible clindamycin resistance due to erythromycin. Ito yung ganun, yung principle ng D-test. Maalala nyo? That differentiate clindamycin resistance among S. aureus. That has D pattern. So erythromycin causes what? Inducible resistance to clindamycin. Ito, the beta hemolysis is produced by group A streptococcus seen on the surface of what? On the surface of sheep blood agar plate is primarily the result of what? Of streptolysin what? HBMCOTS. Ano? S. Correct. Surface hemolysis siya. Why? Because oxygen labile ba siya or stable? Si S ba, oxygen labile or stable? Oxygen stable siya, kaya surface hemolysis. Si O naman, subsurface hemolysis, hindi siya sa surface. Why? Because oxygen labile siya. Pero sinong mas antigenic? Sinong antigenic? O or S? Si O, streptolysin O. Mas antigenic siya. Ito, which of the following organism is able to hydrolyze sodium hypurate to benzoic acid and glycine? A. S. agalaceae, B. E. fecalis, C. S. pneumoniae, D. S. mutans. Ano sagot nyo? A. Yes. Sino sila? Jejuni, Listeria, Gardnerella, S. agalaceae. They are hyporate. They can hydrolyze sodium hyporate to benzoic acid. Ito, optocin sensitivity is used to differentiate what? Letter A. Hegemony from other alpha hemolytic. Ano yung zone niya? Yung susceptible zone niya? Ito. Si bacitracin, toxo A, o other term for toxo A, ang susceptibility niya is 10 millimeter. Susceptible zone niya, 10 millimeter. Take note that susceptible si S. pyo. Si optocin naman, toxo P. Ano na yung chemical name niya? Ethyl hydrocuprin hydrochloride or optocin or toxo P. 14 naman siya. Pa-memorize na lang to. 14 mm. Susceptible si S. pneumoniae. Streptococcus pneumoniae. Sa furazolidon naman or other term is lyostaphin, in which susceptible si S. aureus. Ano ang susceptible zone niya? Greater than 15 mm. Kapag novaviocin naman, take note, resistant si saprophyticus. The other non-coagulase staph are susceptible 16 mm. Then oxacillin and cefoxitin naman, susceptible zone niya is 20 mm. Pa-memorize na lang. Ito. In addition to Enterococcus fecalis, which other streptococci will grow in 6.5 salt broth? A. Group A streptococci, B. Group B streptococci, C. S. pneumoniae, D. Group D non-enterococci. Letter C. Group B streptococci. Ito. Necrotizing fasciitis is a serious infection associated with what? Ano sagot nyo?
B.
B.
Payo.
Yes, payo. Pero paano pag itong tanong nila? What type of necrotizing fasciitis caused by pyogenes? Anong type niya? Kasi may type to eh.
Flesh
Type-
eating.
Type 2 or yung flesh-eating bacteria. Yung type 1, polymyobic, that there is a combination of anaerobes and aerobes or mixed infection usually seen in DM, diabetic mellitus. Tapos yung type 3 naman is the gas gangrene. Causative agent, yun is yung sila Clostridium. So there are types of necrotizing fasciitis. Type 1, type 2, and type 3. Polymyobic in nature, fresh-eating bacteria, and gas gangrene formation. Cirrus-colored sputum in case of lobar pneumonia is characteristic of which of the following possible etiologic agent?
Three.
Letter rust-colored sputum.
Yung mango.
Yun, letter C. Si Streptococcus pneumonia. Number 40. A gram-negative Diplococcus isolated from the middle ear aspirate grew on a chocolate agar but was inhibited on a Martin Lewis agar. The organism failed to produce acid from glucose, maltose, lactose, and sucrose. It was DNA positive and is most likely what? Bacteria. A. And gonorrhea. B. And meningitis, and lactima, and M.
D.
catarrhalis. Letter D. Kasali sila sa what? Asacrolytic. Yung mga hindi nila kayang mag-utilize ng glucose. Asacrolytic organism. Ito sila, pamemorize ako, si Acinetobacter ewophi, Morganella, Oligella, and alkaligenes. They don't utilize glucose as saccharolytic. Ito, sikat to, clay-like consistency on top. Letter C, Neisseria elongata. Again, clay-like consistency on top, letter C, Neisseria elongata. I-eliminate mo na si N. gonorrhoeae kasi hindi naman siya nagtatrive sa BAP, sa cup lang, pati Martin. The correct answer here is Neisseria elongata, clay-like consistency on top. Kapag serrated confluent, the growth P. aeruginosa, snowflakes on BG agar, Salmonella, Hakipak colonies, Moraxella, brittle crumbly wrinkle, Nocardia, battery creamy yellow, SR rus, smooth, glistening, and clay-like, and elongata. Ito, Waterhouse-Friderichsen syndrome is associated with what? Ano? Is associated with letter A, autoimmune response to group A strep, B, septicemia from Neisseria gonorrhoeae, C, adrenal hemorrhage.
Letter C.
Letter C. Noon, hindi ko ito nakuha kasi napagbaliktad ko si Fitzhugh syndrome, which is the disseminated form of Fitzhugh-Curtis syndrome, disseminated form of N. gonorrhoeae. Kaya wag niyo din pagbaliktarin. Waterhouse-Friderichsen syndrome is caused by adrenal hemorrhage from Neisseria meningitidis, or what we call the Bright's disease. Adrenal hemorrhage from Neisseria meningitidis, or Bright's disease, or syndrome. Si Fitzhugh-Curtis syndrome naman, septicemia from Neisseria gonorrhoeae. Septicemia from Neisseria gonorrhoeae. Huwag niyong pagbaliktarin. Gram-negative diplococci are isolated from a female genital tract specimen. Which of the following is the most appropriate action? A. Report Neisseria, gonorrhea isolated and perform susceptibility testing. B. Report gram-negative diplococci and estimate the number of neutrophil present. C. Report normal flora isolated and discard the specimen. D. Plate specimen on blood agar. Ano sagot niyo? Mali na si blood agar kasi possible na may gonorrhea yung specimen. So why would you culture it on blood agar? Hindi naman siya nagtathrive dun. So mali na to, letter D, mali. The correct answer here is what? Letter B. You have to report the gram-negative diplococci and estimate the number of neutrophil. Wag yung i-report right away na Neisseria gonorrhoeae isolated. No, wag yung i-report right away. Kasi take note, female genital tract. Which is in contrast kapag sinabing male genital tract, kapag nakakita ka ng isang gram-negative diplococci lang, i-report mo na agad na gonorrhea infection. But not in the case of female. Kapag female genital tract, you should don't report Neisseria gonorrhoeae right away. Just report the amount of gram-negative diplococci and estimate the number of neutrophil present. Ito, sabi dito. Positive smear in symptomatic male already diagnostic for gonorrhea. Kahit isa lang. Already diagnostic for gonorrhea. For CLAP. 43. Which organism are plump cocobacilli that may resist decolorization and may be mistaken for Neisseria? Resist decolorization and may be mistaken for Neisseria species. Sino to? No. Letter B, Acinetobacter species. They resist decolorization and may be mistaken for Neisseria species. Ito. An isolate of E. coli recovered from stool of a patient with severe bloody diarrhea should be tested from what sugar before sending to reference laboratory. Ano sagot nyo?
Sorbitol.
Sorbitol. Anong kulay niya? Colorless. Ito. It produces a toxin that is similar to Shiga toxin produced by Shigella dysentery. Ano sa letter B? The VTEC or verotoxin E. coli. Verotoxin E. coli or verotoxic E. coli or enterohemorrhagic E. coli. Take note, Shigella-like toxin. Paano kapag Shigella-like infection? Magkaiba ang kapag Shigella-like toxin, siya si VTEC or EHEC. Kapag Shigella-like infection, siya si EYEC or entero-invasive E. coli. Entero-invasive E. coli. Paano kapag cholera-like toxin? Sino siya?
ETEC, sir?
ETEC, yes, correct. Enterotoxigenic E. coli. Take note nyo yung stool niya. Kapag ETEC, profuse watery stool. Kapag VTEC, kapag EYEC naman, yung stool niya may presence scan of neutrophils, mucus, and red blood cell. Kapag ganun naman, kapag enteropathogenic E. coli or EPEC, which is non-invasive toxin, it produces what? Watery diarrhea with mucus but no blood. Watery diarrhea with mucus but no blood. Ang tatandaan nyo kay VTEC, yung sakit na HUS. HUS. Hemolytic uremic syndrome, HUS. Ito, 46. Which single test separate Klebsiella oxytoca from Klebsiella pneumoniae?
Indol.
Correct, indol. Ito, incubation of which of organism at 25 degree produces a characteristic yellow pigment? What's your answer?
Sakazaki.
Correct, Sakazaki. Wait lang. So, a gram-negative bacilli were isolated on Makongi agar from a urine specimen. Biochemical result as follows. Indole negative, glucose acid gas produce. So glucose acid and gas produce, urea positive, H2S positive. Correct answer is what? Proteus vulgaris. Ito naman, which genera of Enterobacteriaceae are usually non-motile at 36 degrees?
A, sir.
A. Klebsiella. It's not non-motile, it's motile. Check ko ulit. Motile to at 36 degrees. Yes. 51. Which of the following organism is often confused with Salmonella species biochemically and on treated media?
Citrobacter.
Yes. Meron tayong tinatawag na Salmonella tribe that you should know how to differentiate Citrobacter from Salmonella kasi nako-confuse daw sila. Ito daw. Si S. fundi, H2S positive siya. Salmonella, I know, it's H2S negative. Sorry, mali. H2S negative to. H2S negative. Tapos, 70% of Citrobacter, especially fundi, as dehydrolyzed urea. Then, I know they are both H2S positive. Sorry. Lito ako sa LDC. LDC negative si S. fundi, tas si Salmonella, LDC positive. Both H2S positive, Salmonella and Citrobacter. KCN positive si Citrobacter fungi, then si Salmonella, KCN negative. Salmonella fails to hydrolyze urea. Ito. In grouping Shigella organism, agglutination by group T antisera indicates that species is what? Letter D, Shigella sonei. Kapag group A naman, Dysentery, Flexneri, B, si Boy D, C, and si sonei is D, group D. Now, the following reactions are true for Pseudomonas aeruginosa except what? Ano sagot niyo dito?
C, sir.
C. Nag-grow siya. 42 degree nga nag-grow siya eh. 37 degree pa kaya. The correct answer here is no growth at 37 degree. Pseudomonas aeruginosa, oxidase positive. B, triple sugar iron slant with an alkaline, no change reaction, and growth at 37 degree, and D, production of bluish green pigment. Nagkamali ata ako ng pag-bold nun. Ito ata na bold ko dun sa answer key. But the answer here is letter C. No growth at 37 degree. Ito, it is characterized by fever, diarrhea, and abdominal pain. Also can cause acute mesenteric lymphadenitis, which may present clinically as appendicitis. Ano sagot nyo?
Enterocolitica, sir.
Yes, correct. Enterocolitica. Yeah. Ito. A presumptive identification of Vibrio cholerae was made after stool specimen from several subjects grew clear colonies on McConkey agar and yellow colonies on TCBS agar, which key tests would help eliminate both Aeromonas and Pisciamonas. Ano sagot niyo?
Oxidase motility, sir.
Oxidase motility?
D.
Letter D. Why? Kasi oxidase, they're all positive. Kasali sila sa sipon or sipap? Sipap. Oxidase positive. Si Vibrio, Aeromonas, Pisciamonas. So, hindi mo di-differentiate using oxidase test ang Vibrio, Aeromonas, and Pisciamonas. Saan? Sa sodium requirements. Negative tong dalawa, Aeromonas and Pisciamonas. Kapag mannitol naman, negative si Pisciamonas. So, correct answer here is letter D. Ito, which of the following bacterial isolates has the following reaction? Negative in MAC, non-motile, negative in nitrite, and no growth in BAP. The answer here is letter A for the telapertussis. Ito, a small gram-negative Coccobacillus recovered from CSF of a 2-year-old child have the following results. Again, CSF of a 2-year-old child. May idea na kayo dun. The results was indole positive, glucose positive, X factor positive, V factor positive, urease positive, sucrose lactose negative or zero, hemolysis zero. Ano sagot nyo?
Haemophilus influenzae.
Yes, Haemophilus influenzae. Basta yung may para hemolyticus, para influenzae, they only required what? V factor. Requirement lang nila is V factor. Pero kapag hemolyticus, influenzae, yung mga walang para, except for Aprophilus, they both required X factor and V factor. Si H2-Crei naman, si Haemophilus 2-Crei naman, it requires what? It only requires V factor. Only requires V factor. And take note, kahit wala to, sinabi dito na 2-year-old child. Meningitis, probably meningitis to. CSF of a 2-year-old child. H. influenzae. Which group of tests best differentiate Helicobacter pylori from Campylobacter jejuni? A. Catalase, oxidase, and gram stain. B. Catalase, oxidase, nalidicylic acid sensitivity. C. Catalase, oxidase, and cephalotin sensitive. D. Urease, nitrite, and hipurate hydrolysis. Ano sagot nyo?
D.
Letter D. Why? Urease positive si Helicobacter, while si Campylobacter jejuni is negative. Sa nitrite naman, positive si Campylobacter sa nitrite, negative si Helicobacter. And so with hipurate hydrolysis, di ba may mga organism yung ng hipurate hydrolysis positive? Si Agalactiae, si Listeria monocytogenes, including Campylobacter jejuni, which you can differentiate it with Helicobacter pylori, which Helicobacter pylori is a hipurate hydrolysis negative, while Campylobacter jejuni is positive. Ito. Urease negative si jejuni, positive si pylori. Nitrite positive si jejuni. H. pylori is negative. Hipurate naman positive. H. pylori is negative. Ito. Which Haemophilus species is difficult to isolate and recover from a genital ulcers and swollen lymph nodes? Ano sagot nyo?
Haemophilus gaprae.
Yes.
B.
Sakto. Ano siya? Soft chunker or hard chunker?
Soft.
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 Correct. The optimal specimen for recovery of Bordetella pertussis is what? A. Anterior nares swab, expectorated sputum, C. nasopharyngeal swab, D. blood. Ano sagot nyo?
C.
C. Yes. C. these are the organisms that could aid in the recovery of hindi, itong mga organism na to, they are recovered in nasopharyngeal swab, H. influenzae, and meningitis, Bordetella pertussis, and S. aureus. Which of the following is optimal clinical specimen for the recovery of L. haemophila? Sagot nyo? Letter B. A fastidious gram-negative bacilli was isolated from a case of periodontal disease, which upon dark field examination was noted to be gliding motility. Uy, gliding motility. Ano sagot niyo dito?
A.
Yes, correct. Ito, smooth gray colonies showing no hemolysis are recovered from infected cat scratch on blood and chocolate agar but fail to grow on McConkie agar. The organisms are gram-negative pleomorphic rods that are both catalase and oxidase positive and strongly indole positive. The most likely organism is what?
B.
B.
Yes, Pasturella. Ito, which of the following species of Bacillus is non-motile?
C.
Anthracis.
Correct. Ito, both are catalase, Sirius and Anthracis, positive. Lecithin is positive siya. Motility naman, non-motile. Again, non-motile. Kasi napagbaliktad ko to. Non-motile. Si B. Sirius naman is motile. Sa hemolysis on BAP, gamma or non-hemolytic si Anthracis and si Sirius is what? Beta hemolysis. Again, beta hemolysis. Ito, which of the following is are true about Bacillus anthracis? A. Non-hemolytic, B. Gamma hemolytic, catalase positive, D. All of the above. Sagot?
All of the above.
Correct. Ito, resembles medusa head. Beta hemolytic and smoothly swarming. Ano sagot nyo?
C.
C.
D.
B.
D. Sino sumagot ng A? Sino sumagot ng A? May sumagot ba? Wala? Walang sumagot ng A? Correct answer here is Clostridium septicum. Medusa head din siya. And beta hemolytic. At just so you know, hindi lang si Proteus ang may smoothly swarming, but siya din. Si Clostridium septicum, medusa head, nire-resemble niya si Bacillus anthracis. Medusa head, beta hemolytic din siya, and smoothly swarming. Clostridium septicum. Letter A is the correct answer. Kasi kung kung yung tanong is kapag medusa head lang, then go for Bacillus anthracis. Pero meron kasi siya beta hemolytic and smoothly swarming, then si septicum yun. And then kapag yung tanong din nila, kapag smoothly swarming lang din at walang medusa head at beta hemolytic, then ang tamang sagot is si Proteus. But si septicum, Clostridium septicum, resembles medusa head, beta hemolytic, and smoothly swarming. Ito. The term palisading, picket fence, and Chinese letter describe the common arrangement of the cells. Baka may sumagot sa inyong letter C, pero okay lang yun. But the correct answer is letter A, Corynebacterium. Ito. Cysteine thyroid blood agar, CTBA, are recommended for the what? For isolation of what? A, C. diphtheria. Yes, letter A. Ito, which two tests can be used to differentiate C. diphtheria from other Corynebacterium? Corynebacteria. A. Halo on thin steel and nitrite test. B. Halo on thin steel and urea hydrolysis. C. Black on thin steel and esculin test. D. Black on thin steel and nitrite test. Ano sagot nyo? Letter B. Hilo siya sa tin steel, and take note na urea passive hydrolysis din siya. Letter B. Ito. Which of the following is incorrect for L monocytogenes? A. Camp test positive. B. Catalase negative. C. H2S negative. D. Motile. Again, the incorrect for L monocytogenes. Ano sagot nyo? Letter C. H2S negative. Ito, the production, baka alam nyo to, the production of H2S is one characteristics used to differentiate which of the aerobic gram-positive bacilli.
B.
B.
Erythropoiesis. Yeah, tama. Lang guys, may double check lang ako. Kaya naman pala. Sorry. Wait lang. Edit ko lang to. H2S positive is incorrect for L monocytogenes kasi H2S negative siya. Kasi dito, ang nagpo-produce lang ng H2S for aerobic gram-positive bacilli is the erythropoiesis, letter B. Nocardia can be differentiated from actinomyces based on what?
B.
Letter B, yes. Letter B, nocardia being an obligate and aerob. The partial,
B,
ano? Letter D, nocardia being the catalase positive. Nocardia can be differentiated from actinomyces based on what? The acid-fast staining reaction of actinomyces. Dapat the partial acid-fast staining reaction of nocardia, hindi actinomyces. Kasi sa actinomyces, it's not hindi siya partial acid-fast. See, nocardia yun. Kapag nocardia being an obligate anaerobe, it's not, nocardia is not an obligate anaerobe. It's actually aerobic. See, actinomyces ang obligate anaerobe. The production of sulfur granules in cases of nocardiopsis. Same lang din sila. We cannot differentiate them, the two. Pero ito, letter D, nocardia being catalase positive. The correct answer here is letter D. Nocardia can be differentiated from actinomyces based on nocardia being the catalase positive. Ito, oxygen requirements, aerobic si nocardia and aerobic si actinomyces. Acid-fast stain positive si nocardia and non-acid fast stain negative si actinomyces. Catalase positive, negative. Positive si nocardia, negative si actinomyces. Urease positive si nocardia, negative si actinomyces. Sulfur granules positive. They are both positive. Ito, sputum samples may contain a normal flora, other contaminating microorganism. So sample must be blank. A, digested with an acetyl-L-cysteine. B, decontaminated with 2% sodium hydroxide. C, digested with trisodium phosphate. D, decontaminated with ditriotratol. Ano sagot niyo dito? Take note, digestion is different from decontamination. We decontaminate in order to remove the contaminating microorganism. We digest the sputum or the sample to remove the the mucolytic agent. Just the example is the mucus. No, the correct answer here is B, decontaminated with 2% sodium hydroxide. Hindi pwede digested. Kasi ang tinatanong is what to do in order to decontaminate a microorganism in a sputum sample. Decontaminate it with 2% sodium hydroxide. Letter C, digested with, the correct answer is letter B. Letter C, digested with trisodium phosphate. Trisodium phosphate is not actually a decontaminating solution. Same with 2% sodium hydroxide. Tama siya sana, kaso dapat decontaminated ang ginamit instead of digestion or digested. Then letter D, decontaminated with dithiotritol. Mali din. But the correct answer here is to contaminate it with 2% sodium hydroxide. Now, usually, we combine 2% hydroxide with N-acetyl L-cysteine in order for them to act as, in a combination, they act as a they digest and decontaminate. Then, si dithiotritol naman, in combination with trisodium phosphate, para at the same time, nagde-decontaminate sila and nagda-digest. Ito, a sputum smear reveals an average of 5 acid-fast bacilli per 1,000 field. According to CDC guidelines, report this result as what? Alam niyo ba yung sagot dito? A sputum smear reveals an average of 5 acid-fast bacilli per 1,000 field. According to CDC guidelines, report these results as what? A, positive, negative. B, positive. C, 2+, D, 3+. Ano sagot niyo?
B, sir. B.
Ano? B?
B, sir.
Letter D.
Letter D. Ito, may tinatawag tayong CDC method of reporting AFB. And meron din tayong tinatawag na AFB grading or national standard. Pa-memorize tong AFB. Mas tawag dun, i-familiarize nyo na yung sarili nyo dito. Pero mas priority nyo pa rin yung AFB grading national standard or local standard. Kapag 0 per 100 field, again 0 per 100 field, negative. Kapag 1 to 2 per 300 field, positive or negative, then i-check mo yung second slide niya. Kapag 1 to 9 per 100 field, then that's 1+. Kapag 1 to 9 per 10 field, that's 2+. Kapag 1 to 9 per field lang, that's 3+. Then kapag greater than 9 per field, that's 4+. Ito, AFP grading national standard naman, 0 kapag no AFP seen 300 fields. Pansin nyo, 300 fields muna kinakount bago mag-report ng 0. Kapag plus naman, plus N, 1 to 9 AFB seen per 100 field. Positive 1, 1 plus naman, 10 to 99 AFB seen per 100 field. Kapag 2 plus, 1 to 10 AFB seen per in at least 50 fields. Kapag 3 plus, greater than 10 AFB seen per field in at least 0 fields. 0 field. Sorry. 75. Which of the following groups is characterized by a negative reaction for heat-stable catalase? Ano sagot niyo dito?
A, sir.
Letter
A po.
Lahat ng M tuberculosis complex, they are heat-stable, catalase negative. Ito, mga M tuberculosis complex, pa-memorize ako nito. MTB, M. bovis, M. africanum, M. capri, caneti, microti, and pinipidi. Ito, acid-fast staining of a smear prepared from digested sputum showed slender, slightly curved, beaded red mycobacterial rods. Growth on Middlebrook 7H10 lands produce buff-colored colonies with a serpentine pattern after 14 days at 37 degrees. Niacin and nitrate adduction tests were positive. What is the most probable presumptive identification?
B.
A.
Letter
A.
A.
Mycobacterium tuberculosis. Nilagay ko na lahat dito. Ito. Which of the following are true about the gold standard used in the diagnosis of leptospirosis? 1. Microscopic agglutination test. 2. Serum antigen killed leptospira. Positive agglutination. 3. Microscopic agglutination test. 4. We use serum plus antigen live leptospira. Then there will be a positive agglutination under the dark field microscopy. Ano sagot niyo? Which of the following are true about the gold standard used in diagnosis of leptospira? Ano yung gold standard? It's a microscopic agglutination with the use of what? Serum plus the antigen, which is the live leptospira, and the positive result is agglutination under dark field microscopy. The correct answer here is 3 and 4, letter D. Ito, rickettsia are negative obligate intracellular. Intracellular bacteria include the genera of Rickettsia, Ehrlichia, Coxiella, and Rockalimea. All of the following are transmitted by ticks except what? 1. R. Rickettsia. 2. R. Prowazekii. C. Ehrlichiosis. 4. R. Tsugamusi.
D, sir.
Answer here is letter D. Si R. Tsugamusi at saka si R. Prowazekii. They are not transmitted by ticks, instead by mites siya. Ito lumabas to noon. Kaso yung tanong, in-identify niya as, yung characteristic niya is obviously si Trachomatis. Kaso may word lang na nagpalito. Sabi niya kasi, but susceptible, but resistant to sulfonamides. At sa libro, sabi dun that si Trachomatis is actually not, it's actually susceptible to sulfonamides, not resistant to sulfonamides. Pero the rest ng binigay given na characteristic niya, tinutukoy niya talaga si Trachomatis. Ito, an obligate intracellular organism with glycogen-containing inclusion but susceptible to sulfonamides. Sagot dito is si Trachomatis. Si Trachomatis. Ito, palitinibol ko na siya. Si Trachomatis has what? Round avascular inclusion, morphology, and bodies. It has glycogen-containing inclusion and susceptible siya as compared sa dalawa, resistant siya. And the diseases is trachoma, LGV, and inclusion conjunctivitis. Sa tuar, strain of chlamydia are associated with which type of infection? Ano ang meaning ng tuar? Taiwan respiratory system. Yes, sakto. The correct answer here is what? Letter B. 81. An agent of sulfongitis, postpartum fever in females, and it produces large fried egg-like colonies. Again, large fried egg-like colonies. Ano sagot nyo?
A, sir.
Letter A. Correct. Si ureaplasma naman, small fried egg colonies. And it uses what? It uses urea. While si M. humanis, si Mycoplasma humanis, it uses what? Arginine. Protein arginine. Si Ureleticum, it uses urea. And small fried egg appearance, si M. humanis is large fried egg appearance. Ito. An organism that exhibits breadcrumb colonies, spindle ship rod, greening on exposure to air, and fluorescent chartreuse. Uy, greening upon exposure to air and breadcrumb colonies. Ano sagot niyo dito?
C, sir.
C. Fusobacterium. Fusobacterium. Ito. What is the predominant indigenous flora for the colon? A. Anaerobic gram-positive non-performing, anaerobic gram-negative non-performing, aerobic gram-negative non-performing bacteria, aerobic gram-positive performing bacteria. Ano sagot nyo?
B.
Letter B. Kasi pag spore-forming na yan, hindi na siya normal flora. So the predominant indigenous flora of the colon is letter B. Anaerobic gram-negative non-spore-forming bacteria. Kaya nila bacteroides. Ito. A tube of a semi-solid medium that contains resazurin appears pink. What does this indicate?
C.
C.
The presence of what? Of oxygen. Si resazurin, may dalawa eh. Resazurin and methylene blue. They are what? They are a redox indicator. Redox indicator nila. Mag-change sila if there is a presence of oxygen from pink. Si methylene blue naman, blue kapag may oxygen. Kapag colorless or without oxygen, si resazurin colorless siya. Kapag without oxygen, si resazurin is colorless. Kapag without oxygen naman, methylene blue, colorless or white. Again, colorless or white. Ito. What is the reagent for sniff or whiff test? A, 10% potassium hydroxide, sodium hydroxide, hydrochloric acid, ethanol. Letter A. Whiff test na lang, wag sniff. Upon adding 10% KOH, ano yung maamoy?
Fishy amay.
Yeah, fishy amine. This is true in what? In bacterial vaginosis, or anong organism yun?
Gardnerella.
Gardnerella vaginalis. Ngayon, guys, kung tatanungin, causative agent for bacterial vaginitis, ano sagot nyo? Kapag bacterial vaginosis, it's Gardnerella vaginalis. Pero kapag bacterial vaginitis, magkaiba yun sa vaginosis. May idea kayo? The causative agent of bacterial vaginitis is mobilincus, bacterial vaginitis. Pero mas sikat si bacterial vaginosis or Gardnerella vaginalis. Ito, in bacterial analysis of water, the confirmed test consists of what? Ano sagot nyo dito? Actually, letter C ang sagot dito. Hindi B. Sorry, nagkamali ako ng ganun. Letter C. In bacterial analysis of water, the confirmed test is what? Typical coliform colonies on EMB agar. May tinatawag tayong presumptive test, which is what? Formation within 43 hours of gas in any amount in fermentation tube. Then after presumptive, meron tayong confirmed test. Either you'll make use of procedure using brilliant green bile lactose, where there is a formation and a presence of gas in any amount within 48 hours. Kapag gamit tayo ng endo or EMB, there is a typical colonies that will develop on the plate after what? In the period of 24 hours. Kapag completed naman, formation of acid and gas in lactose broth and the demonstration of gram-negative spore-forming bacilli in the agar culture. There are 3 steps in bacteriological analysis. These are presumptive, followed by confirmed, and completed. Ito, anong sagot niyo dito? All of the following are major steps in the treatment of communities drinking water except what? A, boiling, B, filtration, C, sedimentation, D, flocculation. Ano sagot niyo?
Boiling.
Sino sumagot ng boiling?
B.
Yeah, letter A, boiling. Kasi si boiling hindi siya part or hindi siya part as the steps in treatment of community drinking water. The first step is filtration.
Yung slide po,
Po,
di lumilipat.
yung screen, wait lang. Anong number diyan?
82, sir.
Ay, 82. Okay na ba? Hindi pa. Okay na?
Hindi pa rin po.
Okay nang mag-break muna 5 minutes. Try ko lang to i-restart. Start ko lang. Paano nagloloko?
Sige po, sir.
Naka-stack pa rin?
Yes, sir.
Okay na? Okay na? So guys, okay na ba?
Okay na, sir.
Okay.
Yes, sir.
Pipicturean nyo ba? Or
Sir,
ha? Pipicturean nyo? To. Saan ba tayo nag-glass?
Dun sa
Dito na?
Yes, sir.
Okay. Pero okay na yung ganun? Kita nyo na? Kita nyo na?
Yes,
yes,
sir.
sir.
Ito, boiling. The first step here is the filtration. You need to filtrate the water in order to remove the debris. After filtration, sedimentation. Then after sedimentation, flocculation or coagulation. Then filtration ulit, then chlorination. Nandito. Wait lang. Ang diba, filtration, it removes the larger piece of debris. After filtration, then you perform sedimentation where the water is held into a tank. Then it takes time para magsisettle down yun. Then after filtration, gagamit ka ng flocculation ka na, gagamit ka ng alum potassium sulfate para makoagulate yung smaller debris. And magsisettle sila at the bottom. After that, i-filtrate mo sila. Kukunin mo yung settled na sediments, and then after that, i-treat mo yung water ng chlorine or chlorination. Bali, chlorination is the last step. Walang step na boiling. Ito, a host in which parasite does not develop further to a later stage. However, the parasite remains alive and is able to infect another susceptible host. Ano sagot niyo dito? Letter C. Kapag definitive host ka, you harbor the adult or the sexual stage of the parasite. Kapag intermediate naman, the larval or the asexual stage of the parasite. Paratenic naman, the parasite remains in you, alive, and is able to infect another susceptible host. That's paratenic. Letter C. Ito, what is this tool to preservative ratio? Ano sagot niyo dito? 3:1. 1:3, stool to preservative ratio. Pero pag preservative ratio to stool, 3:1. Pero ito, stool to preservative ratio, letter D, 1:3. Ito, in what type of stool specimen will you find both trophozoite and cyst? Ano sagot niyo? Soft.
Soft.
Now, alam niyo dito yung viability nila. 24 hours viability of the form stool. 60 minutes naman viability of semi-form stool. Then 30 minutes lang viability of liquid stool. Patandaan, 24 hours viability of form stool, 60 minutes viability of semi-form stool, and 30 minutes viability of a liquid stool. So, which of the following are true about polyvinyl alcohol? 1, not a fixative. 2, eco-friendly. 3, normally incorporated with Scoudin's solution. And D, polyvinyl alcohol with Scoudin's solution is used for permanent staining. A, 1, 2, 3, 4. B, 2, 3, 4. C, 3 and 4. D, 2 and 4. Ano sagot niyo dito? Ano sagot nyo dito? Anyone? Ano sagot nyo dito? The correct answer is A. Baka dinisregard nyo yung 1, pero polyvinyl alcohol is not a fixative. It is an adhesive. Magiging fixative lang siya when it is incorporated with Chaudin's solution. Again, si polyvinyl alcohol is an adhesive, not a fixative. So correct lang. Bali ang sagot 1, 2, 3, and 4. Again, magiging fixative lang si PVA kapag na-incorporate with another fixative. But PVA is adhesive in nature, not a fixative. What is in the top layer in the formalin ether? Ethyl acetate concentration. Letter A. Followed by debris, formalin, and the sediments. Ito, what is the specific gravity of zinc sulfate solution used for flotation technique? Ano sagot niyo dito?
C.
Letter C. Letter C kapag correct answer dito is letter C. Letter C kapag soft stool or fresh stool, the estimate specific gravity of soft stool is 1.18. Kapag naman preserved or formalin preserved stool, 1.20. So, ang specific gravity ng zinc sulfate solution used for flotation technique is ranges 1.18 to 1.20. Letter C yun. Number 94. All of the following are true except modified trichome stain. Except A, it uses trichome 10 times the amount used. B, staining time increased to 90 minutes. C, uses 1 to 3% dihydrogen sulfide as decolorizer. D, used to detect microsporidia. Ano sagot niya dito? Nag-except siya. Ano sagot nyo? Letter C. Why letter C? Kasi C uses 1 to 3% dihydrogen sulfide as a decolorizer is used in a modified acid fast for coccygeans. Used in a modified acid fast for coccygeans. Ang correct answer lang, ang tama lang dito is yung A, B, and C. It's true na si modified trichome stain, it uses 10 times the amount used. Greater than that trichome stain. Kaya siya natawag na modified. Now, staining time, nag-increase siya to 90 minutes. Then, modified trichome stain is used for microsporidia. Pero so for coccygeans, ang stain dun is what? Modified acid fast that uses 1 to 3% dihydrogen sulfide as decolorizer. Ito, which of the following statements are true about fecal cultivation? 1. Distinguish whether the infection is due to S. stercularis or hookworm based on rhabdidiform larval morphology. 2. Allow the development of larvae into the filariform stage for further differentiation. 3. Cultures of parasite in association with unknown flora. 4. Parasites are grown as pure culture without bacterial association. 5. Sometimes referred to as coproculture. The correct answer here is what? Letter D, all of the above. Ang fecal cultivation, either it uses what? Pure culture na sinasabi sa 4, and it also uses what? Culture and association with unknown flora. Example nito is what? Meron tayong tinatawag na monozenic culture na gumagamit siya ng E. coli in order for that parasite to survive. Meron din tayong tinatawag na acinic culture na pure culture lang siya. Hindi siya gumagamit ng bacteria in order for the parasite to survive. Example of this is the NM, the Novi McNeil Nicole medium. Then ang example naman ng monozenic culture na gumagamit ng E. coli is the corneal biopsy culture for Acanthamoeba and Naegleria. Ginagamit nila is E. coli for them to survive. Tas yung sometimes yung fecal culture sometimes called the coproculture. Other term for coproculture is a fecal cultivation. Letter D. Ito, blank is used in demonstration and rapid diagnosis of malaria parasite. Blank is used in species identification of malaria parasite. And blank is a white cell layer smear stained with gemsa stain. Blank it uses capillary tube pre-coated with acridine orange and potassium oxalate and read using fluorescent microscope after centrifugation. Ano sagot niyo dito?
A, sir.
Letter A. Correct. Thick smear, thin smear, puffy coats, films, quantitative puffy coat. A. Ito, amount of formalin used in NATS concentration technique. Ano sagot niyo? Letter B. Correct. Paano nga to? Si NATS used for what? It used in cases of low microfilaremia. We use 1 ml blood plus 9 ml of 2% formalin. Then after that, pakitake note the centrifugation time niya, pati yung speed niya. 500 G for 1 minute. Then 1 minute, discard supernatant, then smeared or stained. Ito, directional progressive movement of trophozoites. The cyst has 1 to 4 nuclei with centrally located karyosome and finely and evenly distributed peripheral chromatin. Ano sagot? A. Histolytica. Now, how will you differentiate true amoeba from other amoeba? Si Entamoeba histolytica, sa pag nag-start ng entamoeba, they are true amoeba. Kapag endolimax na or Iodoamoeba buchli, they are other amoeba. They are not true amoeba. Why? Because a true amoeba possess what? Peripheral chromatin. Walang peripheral chromatin si Endolimax nina or Iodoamoeba buchli or the other amoeba. Pero sila Entamoeba histolytica, lahat na nag-start ng word na Entamoeba. They have peripheral chromatin. Ito, primary amoebic meningoencephalitis. PAM is primarily caused by what?
B.
Primary caused by?
B, sir.
B. Yes, sakto. Primary amoebic meningoencephalitis. Take note that primary amoebic meningoencephalitis resembles the gay. Sino nagkakos ng gay? Granulomatous amoebic encephalitis? Si Acanthamoeba castellani. Yes. Si Naegleria fowleri causes PAM, the primary amoebic encephalitis. Si Acanthamoeba castellani causes gay or granuloma amoebic encephalitis. They are the same kasi they both cause what? Acute suppurative infection ng brain or acute suppurative inflammation of the brain. But what's the difference? Anong pinagkakaiba nila? Si Naegleria fowleri, which cause PAM, it is very fatal than the gay. 95% kapag na-acquired ng isang tao dun within 3 to 7 days, mamamatay na siya agad. Which is in contrast with gay, granuloma with granulomatous amoebic encephalitis. Yun yung pinagkaibahan. But since they both infect the brain, then we need CSF in order for them to detect. Ngayon, kung ikaw yung medtech, how can you say that this CSF is infected with Naegleria and this CSF is infected with Acanthamoeba? Take note, kapag nakita ka ng trophozoites sa CSF, trophozoites lang without cysts, then most likely that CSF is infected with primary amoebic meningoencephalitis or with Naegleria fowleri. But if you see both CSF, both cysts and trophozoites, most likely that CSF is infected with gay. Ano pa ang kailangan tandaan sa gay? Si gay din, si granulomatous amoebic encephalitis, it has a high potential that if you acquire gay, most likely may na-acquire ka din, na-infect ka din ng you're already infected with a pathogenic bacteria. A pathogenic bacteria that has the potential to carry a pathogenic bacteria. Anong bacteria yun? It's Legionella. Once na may gay ka, most likely baka infected ka din ng Legionella bacteria. And then si Acanthamoeba species naman, or Castellani, kilala din siya sa amoebic keratitis, or which is implicated with contaminated lens solution, same with Pseudomonas aeruginosa. Yun lang naman. Ito, which of the following would have a double word wrinkled cyst form? Ano sagot niyo dito?
Acanthamoeba.
Acanthamoeba castellani. Correct. Ito, what is the most common form of Blastocystis hominis that is found in clinical stool specimen? What is the... Ano sagot niyo?
B.
Letter B. Yes, correct. Central vacuum form. Ito, there are forms of Blastocystis hominis. The first form is the cyst form, or this form is a most recently described form. It has thick wall responsible for external transmission through fecal oral route. Thin-walled naman, which is responsible for auto-infection. Then the other form is the central vacuole form, or the central body form. Most common form found in clinical stool specimen. Clinical stool specimen. The amoeboid form naman is rarely seen. And then the last form is the granular, which can be seen in the cultures of B. hominis. So these are the forms of Blastocystis hominis: the cyst form, central vacuole form, amoeboid form, and granular form. Ito, which of the following is a flagellated protozoans that attaches to the intestine via ventral sac? A. Cryptosporidium, B. Isospora, C. Strongyloides turcularis, D. Gargalambia. Ano sagot nyo?
B, sir.
D. Gargalambia. Ito, which of the following protozoa have an undulating membrane? 1. Trypanosoma. Ano sagot nyo?
C po.
C. 1 and 4. Dati in all of the above ko to noon, pero mali. 1 and 4. Letter C. Kapag sucking disc si Garcia, pag spiral groove, chylomastix. Kapag undulating membrane naman, Trichomonas, Trypanosoma, Pentatrichomonas hominis. Sila lang. Walang undulating membrane, si Garcia at si chylomastix. Si chylomastix, spiral groove, sucking disc, Gargalambia. Gargalambia sa ito. The hemoflagellate form that is long and slender, undulating membrane is absent, and kinetoplast is anterior to nucleus. May only be seen if a blood sample is collected immediately after transmission. Ano sagot nyo?
A, sir.
The correct answer dito is promastigot. Take note that these forms, si amastigot, promastigot, promastigot, epimastigot, they are found, all forms are found in Trypanosoma cruzi infection. Pero si Trypanosoma brucei, either gambiense or rhodesiense, makikita lang dun is epimastigot and trypomastigot. Then si Leishmaniasis naman, ang makikita lang dun is amastigot and promastigot. Now, what is the infective stage for trypanosomiasis? Infective stage for trypanosomiasis to final host is what? Trypomastigot. Kapag sa Leishmania naman, ang infective stage ng form ang infective stage to final host is the promastigot. Now, si amastigot found intracellularly, primarily in tissue as well as in the CNS. Si trypomastigot naman reproduce and are visible in the peripheral blood. Si promastigot may only be seen if the blood sample is collected immediately. Siyempre, it is infective stage ang promastigot. So, ibig sabihin kapag nakagat ka ng vector, kapag nag-collect ka agad kaagad-agad ng blood, makikita mo yung promastigot. Then si epimastigot naman, found primarily in arthropod vector. Si promastigot, ang infective stage para sa atin. Pero sa vector, ang infective stage sa vector is the epimastigot na makikita lang sa vector. Ngayon, may only be seen if blood sample is collected immediately after transmission is promastigot. Number 5, the winter bottom sign is associated with what?
C, sir.
Letter C, Gambianse. Or the West African sleeping sickness. Ano na nga yung winter bottom sign? There is an enlargement of what? Posterior or cervical lymph nodes. Kapag kirandal naman, there is a delayed in sensation to pain. Sometimes napapagbaliktad ko to. Again, winter bottom sign, enlargement of posterior cervical lymph nodes. Kapag kirandal signs naman, delayed sensation to pain. Si T. cruzi naman, responsible for the disease or for chagoma or small painful reddish nodule on bite site. Chagoma yun. Kapag romanus sign naman, periorbital swelling of edema of eyelid. Romanus sign, periorbital swelling edema of eyelid. Ngayon, si T. cruzi lang yung may capability na kapag chronic ka, mag-enlarge yung organism, yung organ mo. Or enlargement of vital organs if you are chronically infected with what? With T. cruzi. Ito, this nematode is incapable of maturation within the human host and migrate aimlessly causing tissue damage and inflammation. Ito, hindi ko to alam nung nagbo-board ako, pero ang sagot ko, Ganastoma spinigerum, which is correct naman, letter B. This nematode is incapable of maturation within the human host and migrate aimlessly causing tissue damage and inflammation, letter B. Ito, habitat of adult Trichinella spiralis, A. Small intestine, B. Skeletal muscle, C. Blood, D. CSF. Anong sagot niyo dito?
Skeletal muscle.
Yun. Ito, si
A.
yes, A ang sagot. Si skeletal muscle ang naghahabit, doon naghahabit ang Trichinella spiralis in a larval form. Pero kapag adult na, sa small intestine siya. Kaya nga tinatawag siya as what? A phasmid of small intestine. A phasmid of small intestine. Again, kapag si Trichinella spiralis, kapag adult form na, small intestine, pero kapag larva, sa skeletal muscles, di ba? Skeletal muscles. Then, ang tatandaan pa natin kay Trichinella spiralis is the great imitator. Same with syphilis, great imitator din siya. Trichinella spiralis, great imitator din. Again, the adult form is small intestine. Kapag sa larva, skeletal muscles. Ito, Plasmodium species divide through what?
Multiple fissions.
Multiple fission, correct. Ito, which of the following parasite is found in the large intestine? A. Lumbricoides, Ascaris lumbricoides, C. Vermicularis, D. Ano sagot nyo?
B, sir.
B. E. Vermicularis. Si E. Vermicularis is phasmid of large intestine, which it has what? It possesses caudal chemoreceptor. Phasmid of large intestine. Sino yung phasmid of large intestine? Si Trichuris trichuria. Trichuris trichuria, yes. Ito, the worm has characteristic thick cuticle and large uterus and fills the body cavity and contains rhabdiform larva. Ano sagot niyo dito? Sikat na sikat siya as thick cuticle and large uterus. Dracunculus medinensis. Ito, the adult female is characterized by having a barber's pole appearance due to the looping of the whitish uterus around the reddish digestive tract. It causes human eosinophilic meningoencephalitis. Causes cantonensis. Correct. Ito, the zoonotic disease known as clipping eruption, or it forms a tunnel after kapag pag nag-invade siya. Ano sagot nyo?
Braziliensis, sir.
Yep, Braziliensis, letter D. As in visceral larval migrant. Ito, cutaneous larval migrant. Visceral larval migrant. Sino sila?
Toxocara.
The dog and cat ascarid, Toxocara. It is considered among a patient diagnosed to have pulmonary tuberculosis or PTB and not responding to treatment, and among patients suspected to have PTB in Sorsogon. Ano sagot nyo?
C.
Letter C, paragonimiasis. Sakto. Letter C. Ito, sparganosis is due to infection of man by procercoid larva. Ano sagot nyo?
B, sir.
Letter B, dilatum. Diba, ang stages ng development ni dilatum, normally tayo mga adult, once if we are infected with dilatum, tawag dun is diphyllobotriosis. If we are infected as a definitive host, as a definitive host. Pero may times kasi na we act as the secondary intermediate host na instead of the diphyllobotriasis, asparagnosis ang infection natin. Why? Diba? Kapag ang development niyan is, it starts with an embryonated, operculated oval. After that, it will undergo to larval stage. Ang first intermediate stage is the coracidium, which is the infective stage for the copypads or intermediate host. This is the copypads. Then after coracidium, procercoid. Magde-develop siya ng procercoid. It is the infective stage to the secondary intermediate host, which is the fishes. Ngayon, magkaka-asparagnosis tayo kapag once kinain natin yung copypads. Yung copypads which contains the procercoid. Ngayon, tayo as a human, magka-act tayo as the secondary intermediate host. Dun, ang tawag sa infection na yun is asparagnosis. Ngayon, kung for example, kakainin man tayo ng layon, then ang tawag dun sa layon na yun is ang infection na yun is diphyllobotriosis. Na supposedly dapat yun ang infection natin kapag sinusunod yung ganun. But there are cases na may nakakakain tayo ng infected na intermediate host, ng first intermediate host. That's asparagnosis. Sparganosis, letter D. Ay, letter B, sorry. Letter B yun. Dilatum. Ito, host classification of a human infected with hydatid disease caused by ignorlosus. The correct answer here is the accidental intermediate host. Na-bold ko ata yung ganun B, pero the correct answer here is accidental intermediate host. Ito, which tapeworm proglotid makes its way across the fecal specimen by doubling movements and seen under the microscope to have a numerous branching resemble those of tree. Ano sagot nyo?
Saginata, sir.
Saginata. Tree-like dichotomous uterine branches, T. saginata, 15 to 20 uterine branches. Kapag finger-like or dendritic branches, T. solium or 7 to 13 uterine branches. Ito, it causes alveolar hydatid disease which is fetal form of echinococcosis. Ano sagot nyo?
Multilocularis.
Letter B, Echinococcus multilocularis. Again, fetal form of echinococcosis. Echinococcus. Infection with conorchis or opistorchis may result from eating a raw or undercooked. Ano sagot nyo?
D.
D. Freshwater fish. Ito, what is schistosomium? Mali ko to noon. nung compre din namin? Ano sagot nyo?
Cercaria without a tail, sir.
Yes, Cercaria without a tail. Sagot ko is free swimming Cercaria. Letter D. A diagnostic characteristic of the egg of S. japonicum. Ano sagot nyo? Letter D.
D, sir.
Which of the following schistosome eggs may recovered in rectal biopsy? Sagot?
C, sir.
C. Yes. Ito lahat. C. Ito na. What is the optimal temperature and incubation time before a fungal blood culture is reported? Ano sagot nyo?
B, sir.
Letter C. 30 degree to 21 days. Ito, which of the following spores are involved in sexual reproduction? Ano sagot nyo dito?
A, sir.
A. Basta may spores, yung last niya, they are sexual reproduction. And a fungal or a mycosis that has this kind of reproduction, they are called perfect fungi or perfect fungal perfecti. Kapag conidia naman, they are asexual reproduction. Ito, a dermatophyte which large multicelled club-shaped smooth-walled macroconidia, single or in cluster 2 to 3, and a microconidia not formed. Ano sagot niyo dito?
Flocosum.
Flocosum.
Flocos.
Mahilig siya sa club. That's why flocosum. Si M. canis, M. gypsum, M. diodoni, they are and including E. flocculosum and the trichosporon. Yeah, trichosporon. Is it trichosporon? Trichosporon. Trichophyton, they are under dermatophyte. Again, dermatophyte, they are cutaneous mycosis. Magkaiba yung superficial mycosis or tawag dun, superficial mycosis na there's no cellular response. Ang sikat na sikat dun is si Malassezia furfur. Sikat siya sa what? Spaghetti and meatballs appearance. Si dermatophyte naman, ito sila. Si microscope, they has a strong affinity to what? To keratinized tissues like skin, nails. Dun sila nag-infect. Si M. canis, this is zoophilic. Si M. gypsum, tawag dun, zoophilic si canis. Si Odoni naman is anthropophilic. Pero siya lang ata yung positive siya sa nagpo-fluoresce siya sa ganun sa fluorescence, nagpo-fluoresce siya as compared to canis and gypsum. Si sagot dito is E. flocosum. Again, kapag dermatophyte, ang characteristics niya is dry, horny, pit-like lesions or derm scales. Dry, horny, pit-like lesions. They are not horny, sexually, pero horn, they have horn. Dry, horny, pit-like lesions. Number 25. A few smooth fold cigar-shaped microconidia and spherical often in a grape-like cluster microconidia. Positive in yeast and hair beating test. Ano sagot nito?
Mentagrophytes.
Mentagrophyte, correct. Si Sean Laney, sikat siya sa what? Sa Favic chandelier. Tandaan na lang Favic chandelier. Yeah. Trichophyton, Sean Laney. Ito, which of the following is a eukaryotic organism or mycetoma or a granulomatous tumor of subcutaneous tissue? Again, eukaryotic organism, fungi. Ano sagot nyo? 1 and 2. Eukaryotic fungi, 1 and 2, extraphala and pseudostereoboidy. Kapag actinomyces endocardia, they can cause mycetoma, but in a bacterial form. They are what we call actinomytic form of mycetoma. Diba yung eukaryotic form of mycetoma? Hindi sila kasali, actually they resembles the fungi, pero hindi sila kasali because hindi sila nag-grow from a primary culture media of mycosis, which is the Sabaros dextrose agar. Hindi sila nag-grow dun. Siguro kung nag-grow sila dun, siguro they are considered as fungi, pero hindi. Ngayon, eukaryotic fungi causes mycetoma is extraphala and Scleria boydii. Ito, yeast phase of a large round, thick-walled spherules with endospores observed in a tissue and agent of valley fever. Ano sagot niyo dito?
A, sir.
A. Coccidioides immitis. Si Blastomyces dermatidis, South African blastomycosis, ang ganun niya, ang yeast phase niya sa human, ang appearance niya is it's smooth or the broad base. Si Paracoccidioides naman, sikat na sikat siya sa what? Sa appearance niya as a mariner's wheel or Mickey Mouse cap. Ito, encapsulated yeast cells in bird and bath droppings, demonstration of the capsule by India ink stain with a gram stain of classic starburst pattern. Urease negative, inositol positive, but nitrite. Ano sagot niya?
A, sir.
Letter A. Ito, D. Which of the following are true about Candida albicans? Letter D. All of the above. Germ tube positive, cause thrush, vulvovaginitis, onychomycosis, and paronychomycosis. Green color on chrome agar. It is recommended for cultivation of chlamydiospores bearing C. albicans with chlamydiospores production after further enhanced by addition of 1% twin 80, si corneal agar without dextrose. Corneal agar without dextrose. Hindi magta-thrive si albicans kapag with dextrose. Again, corneal agar without dextrose. Ito, letter D. Which of the following organisms are partially or completely inhibited by media containing cycloheximide? Letter D. The protein coat that surrounds the nucleic acid for a virion is called which of the following? Ano sagot nyo?
Capsid, sir.
Capsid, correct. Which of the following swabs cannot be used for specimen intended for viral testing? Sagot nyo?
C, sir.
Calcium alginate, correct. A viral culture that is derived directly from tissue, these cells have a normal number of chromosomes or diploid. Sagot dito is primary cell culture and are permissive for a number of viruses. Kapag established cell line naman, diploid sila, pero mas magagamit mo siya as matagal yung viability niya then sa primary cell culture o mas magagamit mo siya ng matagal. Kapag continuous cell line, dito na pumapasok yung HeLa cells or the cancer cells which is not diploid in nature. Ito, which of the following viral receptor is incorrect here? Si rabies. Why? Dapat sa acetylcholinesterase. Si coronavirus sa Asia. Angiotensin converting enzyme. Si HIV, correct lang to. CXC4 and CCR5 and CD4. Now, CXC4 act as the co-receptor in order for the HIV virus to gain entry in what? In lymphocytes. Ang CCR5 naman, co-receptor with CD4 to gain entry in macrophages. So, correct lang sila. Ang mali dito, si rabies ICAM1. Dapat si rabies acetylcholinesterase. In viral genome replication, eclipse stage, all DNA replicate in the nucleus except parvo, except pox virus. And then take note that all RNA viruses have replicate in the cytoplasm except influenza virus and retrovirus. Ito, hand, foot, and mouth disease is a mild contagious viral infection common in young children. It is characterized by sores in mouth and rash on the hands and feet. Hand, foot, and mouth disease is most commonly caused by what?
A, sir.
A. Coxsackie virus. Now take note that Coxsackie virus is subdivided by two. Coxsackie virus A, which causes herpangina, hand, foot, and mouth. Coxsackie virus B, pleurodina, pericarditis, and myocarditis. Ngayon, kapag tinanong foot and mouth disease lang, it's not Coxsackie virus A. Instead, aptovirus. Kapag hand, foot, and mouth disease, that's Coxsackie virus A. Kapag foot and mouth disease lang, aptovirus. Ito, a 13-year-old was admitted to hospital following a 2-day visit with a relative in the Christmas holidays. Vomiting and diarrhea left the child severely dehydrated. No other members of the family were affected. All bacterial cultures proved negative. A stool sample should be tested for which virus? Ano sagot nyo?
Rotavirus.
Rotavirus, correct. Ito, the name is discovered from enteric cytopathic human organ because the disease associated with these agents was not initially known not until 1967. Newly isolated enteroviruses have been distinguished numerically. Anong sagot niya dito?
Ecovirus.
Ecovirus. Ito naman, dito 3-year-old or for infant type, ito naman adult type. Anong sagot niya dito? Which virus has been implicated? Norwalk, correct. Si Molluscum contagiosum, saang anong member ng virus siya?
Tawag, sir. Pax, sir?
Yes, Pax virus. Ito, si croup, si para-influenza are si respiratory syncytial virus, and metahumovirus. They are responsible in causing croup in pediatric patients. Si filovirus that has characteristic or Shepherd's hook, ano siya?
Marburg.
Marburg virus. Kapag sa parasites?
Manzanella streptocerca, sir.
And
Yes.
Chylomastix. Which of the following viruses causes an acute fever rash and produces disease in immunocompetent children? Ano sagot niya dito? Fifth disease, transient aplastic crisis, parvovirus, letter C. A young father and his 2 small children complained of a rash on the torso of his body. The children had been diagnosed with a chickenpox and confined to their home. The father had experienced chickenpox as a child and he knew he did not have the same rash as children. What is the most likely cause of father's rash? A. Herpes simplex A1 virus, varicella zoster virus, C. Herpes simplex 2, D. Epstein-Barr virus. Ano sagot nyo?
B.
Letter B. Varicella zoster pa rin siya, pero on the second time around, shingles na yung tinatawag dun. Which is magkaiba from first time na na-infect. Kaya magkaiba yung rashes ng bata sa kanya. Ito, influenza A virus undergoes recombination events that produce new strain. This is referred to as what? Is it antigenic drift or antigenic shift?
Antigenic shift, sir.
Yes, correct. Kapag drift, minor lang, changes that occur infrequently. Kapag major changes that result in a novel viral antigens. Ito, si pregnant woman was seen by her obstetrician with a signs of flu-like illness during her second trimester. The NAT tests were ordered for a toxoplasma gondii, rubella, cytomegalovirus, herpes simplex 1 and 2. All test results were negative. However, after delivery, the newborn exhibited signs of an infection and failed to survive. Which virus causes a 90% transplacental infection rate when found in a mother? Ano sagot niyo dito?
B, sir.
B. Actually, matalino sana yung doctor kasi he made use of NAT, which is sensitive and specific, then dun sa conventional na torch test. Kaso siguro ang alam niyang meaning ng torch, yung TO stands for toxoplasma lang, pero hindi niya alam na yung O may meaning yun. O means other form of a transplacental infection. Kapag hindi mo consider as a torch panel yun kapag hindi mo natatest yung other form of transplacental infection, just like HIV, syphilis, and Hep B virus. Kaya confident yung doktor na baka normal delivery yung patient kasi torch niya is negative daw. Pero hindi niya na-complete yung torch. Hindi niya na-test yung other. Kaya yung meaning torch is toxoplasma, then other transplacental infection, then rubella, cytomegalovirus, and herpes simplex 1 and 2. Ito, 148. An outbreak of hepatitis A infection occurred after a meal in a certain restaurant. What is the most probable reason for this outbreak? Ano sagot niyo dito?
A, sir.
Letter A. Sorry, C nilagay ko. Letter ito. letter E. Then, ito na. All of the following viruses can be transmitted by an arthropod vector except letter D. Ito, also known as the breakbone fever. Ano na nga yung breakbone fever? Sino na nga?
Dengue,
Kapag
sir.
dengue fever, correct. Take note that thrombocytopenia with leukopenia and hematocrit level increases in a dengue infection. That's breakbone disease. Sa wakas. Tapos na. Bali sa, gano'n na tayo. Sa HTMLE na tayo. Ito, what type of epithelial cells that found in the linings of alveoli of the lungs and blood vessels?
Simple squamous, sir.
Simple squamous. Correct. Ito sila. Kapag simple squamous, alveoli of the lungs, blood vessels, and endothelium. Kapag simple cuboidal, surface of ovaries, kidneys, kidney tubules. Simple columnar lining of digestive tract from stomach to anus. Pseudostratified columnar lining of a nasal cavity and a nasal sinuses. Kapag stratified stratified squamous naman, both keratinized skin, extra-keratinized skin, non-keratinized mouth, throat, and esophagus. Kapag stratified cuboidal naman, sweat glands, ovarial follicles, and mammary gland or larynx, pag transitional epithelium lining of urinary bladder. Ito, which of the following are not true about the cardiac muscle? Ano sagot niyo dito?
C, sir. B.
Letter B. Take note, si cardiac muscle, it is non-striated. Then, it is not a fusiform cell. Si fusiform lang siya. Ang fusiform type lang is si smooth muscle. Then si smooth muscle, non-striated siya. Si cardiac muscles, both and skeletal muscles, they are both striated. Pero ang voluntary muscles lang si skeletal muscles. Now take note na si cardiac muscles, sikat, meron siyang intercalated disc na tinatawag. Again, cardiac muscle is involuntary, striated, not non-striated, and not fusiform cells, and it composed of intercalated discs. Ito, which of the following is our true symptoms? 1, fever. 2, malaise. 3, pain. 4, hepatomegaly. Kapag tinatawag nating symptoms, this is subjective. Nararamdaman ng patient. Kapag signs, this is objective. That can be graded or that can be measured. Ano sagot nyo? Letter D. Ito, this is due to the transfer of internal heat to the surface or site of injury brought about by increase in blood content. Ano sagot niyo dito?
Color, sir.
Color, correct. Kapag rubor, dati-rati si rubor, color, tumor, and si dolor, sila lang 4 yan. Then diniscover ni Virchow yung functionalia, the destruction of functioning units of tissue. Dati originally 4 lang sila. Then dinagdag lang si functionalia. Kapag rubor, there is an increased rate of blood flow towards the site of injury. Kapag color naman, transfer of heat to the surface or site of injury brought by increase of blood content. Kapag tumor, increased capillary permeability allowing the extravasation of blood fluid, causing localized edema. Dolor naman is the direct damage to nerve ending, and the last one is destruction of functioning units of the tissue. Ito, a type of exudate is characterized by hypersecretion of mucosa with degenerative changes in epithelium. Mucus is a main component of exudate. Anong sagot niyo dito?
Purulent, sir.
Letter C, katarhal. Si purulent naman, characterized by production of large amount of pus or purulent exudates. Si katarhal, take note na hypersecretion of mucosa, characterized by hypersecretion of mucosa with degenerative changes in epithelium. Mucus is the main component of the exudates. Kapag fibrinous naman, characterized by what? Characterized by large amount of fibrinogen and precipitation of fibrin mass. Kapag serous, outpouring of watery or low protein fluid from either the blood serum or secretion of serosal mucosa. Kapag hemorrhagic siya kapag there is a mixture of blood. Ito, blank is the increase in the size of individual cells. Blank is the increase in the number of cells resulting from growth of new cells. Ano sagot nyo?
A, sir.
A. Kapag hypertrophy, there is increase in the individual, there is enlargement due to the increase in the individual size of the cells. Kapag hyperplasia naman, there is enlargement of an organ or tissue due to increase in the number of cells resulting of the new growth. Ito, characterized by the incomplete or defective development of tissue organ represented only by the mass of tissue. Ano sagot nyo? Aplasia. Letter D. Aplasia. Sorry. Kapag Ichnesia naman, complete non-appearance of an organ. Kapag atresia, failure of an organ to form opening. Kapag atrophy, acquired decrease in size of normally developed or mature tissue or organ resulting from reduction in cell size or decrease in total number of cells or both. Number 8, degenerative changes that is characterized by reversible transformation of tissue in a one type of adult cell to another. Ano sagot niyo dito?
A, sir.
Yes, A. It's metaplasia. Dati-dati nahuguluhan yung classmate ko about pinagkaibahan ng metaplasia, dysplasia, anaplasia, and neoplasia. When we say metaplasia, yun nga, there is a one or reversible, which means it is reversible. There is a transformation of one tissue in one type of another of an adult cell to another. Perfect example is the glandular metaplasia. When this is happened in the distal part of esophagus, when that particular part of the organ exposed into a chronic tawag dun, low pH, and then in order for that tissue to thrive, nagta-transform siya from squamous cell to glandular type of cell. Glandular tissue is not an abnormal cell. What makes it abnormal is that supposedly dapat yun nandun is the squamous cells, hindi glandular type of tissue. And then kapag dysplasia naman, there is what we call there is a regressive alteration or tawag dun, there is a variation in shape and the orientation of a cell. It is also reversible. Then sa anaplasia naman, dito, marked by what? Marked by regressive change in adult cells toward more primitive or embryonic cell types, usually a criterion toward malignancy. That's anaplasia. Ito, cytologic picture suggestive but not conclusive of malignancy. Nakuha nyo ba to? Yung number 10? Letter B, class 3. This is based on the presence of malignant cells. This is a grading system in a pap smear, pero obsolete na sila. Pero nakikita ko pa rin to sa mga reviewers ko, kaya na-include ko na. Kapag class 1, there is an absent or atypical or normal cells. Kapag class 2, a typical cytological picture but but there's no evidence of malignancy. Kapag class 3 naman, cytological picture suggestive but not conclusive of malignancy. Kapag class 4, cytologic picture strongly suggestive of malignancy, then class 5, cytologic picture conclusive of malignancy. Ito, a form of necrosis is commonly encountered when arterial supply is cut off seen in anemic ischemic infarction. Ano sagot nyo?
Coagulation, sir.
Yeah, coagulation necrosis. Kapag liquefaction or liquefative necrosis, this is usually happen sa brain when there is a rapid dissolution of enzyme in the brain. Kapag fat necrosis naman, sa pancreas naman siya. When there is a leakage of lipase, and that lipase will metabolize the fatty acids. Kapag caseous naman, this is special type of necrosis na nakikita sa tuberculosis. Ito, sagot niya is letter A. 12, these are the primary changes or signs of death except what? Except, postmortem clotting. Letter C. It is characterized by cooling of the body, occurring at the definite rate about 7 Fahrenheit per hour, and usually important in establishing the approximate time of death. Ano sagot niya? Algor mortis. Ito, which is true about livor mortis. Purplish discoloration is due to stasis and eventually settling into blood vessel. Discoloration disappears upon pressure, reappears upon release, oozing of blood upon incision, and found in the blood vessel. Ano sagot niya dito?
A, sir.
Letter D, 1, 2, and 3, and 4. In livor mortis, you need to know the difference between livor mortis and ecchymosis. Kapag ecchymosis, usually happen siya sa usually happen before death. In contrast siya sa livor mortis na kapag naglagay ka ng pressure sa purplish discoloration, nagdi-disappear, then after release of pressure, kapag na-release yung pressure, nag-reappear. Pero kapag ecchymosis naman, hindi. Plus, oozing of blood upon incision sa ecchymosis, hindi. Then ecchymosis, leakage ng blood, hindi lang sa blood vessel, kundi sa tissue. Ito, found in the blood vessel. Letter D. True about postmortem clotting, except, ano sagot niya dito? Ano sagot niya dito? Letter B. Kapag postmortem clot and antemortem clot, magkaiba sila. Yellow appearance, friable naman si antemortem. Sa sediment niya, usually called chicken fat. Fibrin precipitation entangled, and it has a irregular fashion naman kapag antemortem. Kapag current jelly consistency or rubbery consistency, it's postmortem clot. Antemortem clot, usually granular and not readily detectable. Postmortem clot after death, then antemortem clot is before death. It is the simplest type of marcotome that is used for cutting serial sections of large blocks of paraffin embedded tissue. Ano sagot niyo dito?
Cambridge, sir.
Cambridge. Or what we call rotary. Padwell and Trefol, na-discover noong 1881. Simplest type of, simplest hindi most common. Simplest type of marcotome. This is used for cutting serial sections of large block of paraffin embedded tissue. Kapag rotary naman si Minot, 1885 to 1896. This is most commonly used for sectioning of paraffin embedded sections. Si Adam naman, most dangerous type of microtome, discovered developed 1980s, invented 1789. Si freezing microtome, si Quequet, used to cut undehydrated tissue in a frozen state. Kapag cryostat naman, most commonly used for rapid preparation, -5 to -30 degrees. Sorry, -5 to -30 degree. Ito, it is a type of sliding microtome where block holder is moving while the knife is stationary. Ano sagot nyo?
B, sir.
After a base sledge microtome, when the block holder is moving while the knife is stationary. Si standard sliding microtome, most dangerous siya. Why? Instead of the block holder ang mag-move, yung knife ang mag-move, then stationary si block holder. It is used for base sledge, rotary, and racking microtome. The less concave side is for celluloid embedded tissue blocks and more concave side is for paraffin embedded tissue blocks. Ano sagot nyo?
A, sir.
A, plain concave. Kapag biconcave, both sides used for paraffin embedded tissue block. Then paki-memorize yung these conventional steel knives, paki-memorize yung sizes nila or yung length nila. Plain concave 25 mm, biconcave 120 mm, plain wedge 100 mm. These knives are used to cut blocks for electron microscopy. Ano sagot nyo?
C, sir.
C. Removal of nicks, irregularity, heel to two direction. Ano sagot nyo dito? Honing, hard sharpening, stropping, and polishing. Correct answer dito is A. Kapag stropping naman, removal of bars. And polishing. Two to heel direction naman siya. Ito, it is obtained when the sides of the wedge knife are inclined at an angle of 15 degrees, causing a maximum penetration of tissue and minimizing distortion. Again, perfect angle to. Ano sagot nyo?
B, sir.
B. Kapag bevel angle naman, this is the angle that formed in between the cutting edges. Kapag clearance angle naman, between the blocks and the cutting edges. Bevel angle 27 to 32. Then clearance angle 0 to 15. Ito, the automatic tissue pressing machine, Autotechnicon, performs the following steps. Ano sagot nyo? Except for what? Ano sagot
B,
nyo?
sir.
Except for letter C. Embedding. Why? May mga stations tayo. Kapag station 1 to 2, ito yung sa automatic tissue processing to. Station 1 to 2, 10% formalin. Kapag stations 3 to 6, 3 ascending grades of ethyl alcohol. Kapag station 7 to 8, 2 changes of acetone. Kapag station 9 to 10, 2 changes of chloroform. Stations 1 to 2, 2 changes of liquid paraffin. Again, in automatic tissue processing machine, it performs the following except for embedding manually. Tayo ang gumagawa. Ito, type of biopsy that removes the entire area in question. Ano sagot niyo dito?
Excisional, sir.
Excisional, correct. Kapag fine needle biopsy, it uses small needles, then I aspirate yung bukol, and then smear. Kapag core needle biopsy, it removes the area in question, and then kumukuha siya ng konting amount of surrounding tissue. Kapag excisional naman, it removes the entire area in question, but not in the case of incisional. Kapag incisional, kinukuha yung bukol, tapos, for example, kapag breast, kinukuha yung bukol, and then small amount or large amount of the surrounding tissue, and then the specimen will be sent to lab, that's incisional biopsy. Then kapag sabi ng doktor na cancerous, that's the time na i-excise na yung yung whole na breast. Selected tissue specimen is immersed in a watch glass containing isotonic salt solution, carefully dissected or separated, and examined under a microscope. Ano sagot nyo dito?
B, sir.
Letter A. Si B, squash preparation, kukuha ka ng 4 mm na tissue. And na tissue, then ilalagay mo sa glass, then ipe-press mo with what? Ipe-press mo with the cover slip or another glass slide. And then after that, pwede mo siyang patakan ng stains and then read in under the microscope. Kapag smear preparation naman, si pull apart, types lang siya ng smear preparation. This is useful for preparation for preparing smears of thick secretion such as serous fluid, concentrated sputum, enzymatic lavage samples from the gastrointestinal tract, and blood smear. Ano sagot nyo dito? Letter C, pull apart. Ito, types of smear preparation. Kapag sticking, you have to use of an applicator tip or a platinum loop. Material is rapidly and gently applied in a direct or zigzag line throughout the slides. Kapag spreading naman, the material is transferred in a clean slide and gently spread into a moderate thick film by teasing using an applicator stick. Kapag pull apart, a drop of secretion or sediments upon one slides and a facing it to another slides, and the slides are pulled apart with a single uninterrupted motion. Again, this is useful in the preparing smears of thick secretions such as serous fluids, concentrated sputum, enzymatic lavage, samples from the gastrointestinal tract, and blood smear. Kapag touch preparation naman, special method of smear preparation whereby the surface of freshly cut tissue is brought into contact and pressed on the surface of a glass slides. Ito, all of the following are good characteristics of a fixing fluid except A, it must be cheap, stable, and safe to handle. B, it must inhibit bacterial decomposition and autolysis. C, it must be isotonic, causing minimal physical and chemical alteration. D, it must be it must make cellular component sensitive to subsequent processing. Ano sagot niya dito?
B, sir.
Letter D. Why? What makes it wrong? Ano ang nagpapamali dito? Anong word? It should be insensitive, not sensitive. It should be insensitive, not sensitive. Ito, the amount of fixing fluid should be approximately what? Ano sagot nyo?
A, sir.
It should be approximately A. 10 to 20 times the volume of specimens. Ito, factors that retards the fixation. 1, size and thickness. 2, agitation. 3, presence of blood. 4, presence of fats. 5, cold temperature. All except what? Agitation. Hindi niretards the agitation ang fixation. Hence, it enhances the fixation. Kapag size and thickness, it's true na niretard niya ang fixation kapag larger tissue. Pero kapag smaller tissue lang, the size is smaller, then it enhances the fixation. But the correct answer here is what? 1, 2, 3, 5. 1, 3, 4, 5, letter C. Ito, the microanatomical fixative are those that permit the general microscopic study of tissue structures, and it must never contain what? Kasi ang microanatomical fixative, they are tissue that is subjective to this fixative. They are soon be stained in H and E stains. Ngayon, ang HNE stains, hindi niya ni-stain kapag yung fixative na ginamit mo is what? Ano
A,
sagot nyo?
sir.
Letter A, osmium tetroxide or osmic acid. Number 30. All are true for glacial acetic acid except,
C, sir.
yep, why? It causes tissue to swell dapat. Kina-counteract niya yung action ni mercuric chloride na it causes tissue to shrink. Si glacial acetic acid causes tissue to swell. It fixes and precipitates nucleoprotein, precipitates chromosome and chromatin materials. It causes tissue to swell. It is contraindicated for the cytoplasmic fixation since what? It destroyed mitochondria and Golgi elements of the cells. Ito, which is are true about nuclear fixative? 1. Contains glacial acetic acid. 2. pH less than 4.6. 3. pH greater than 4.6. 4. Must never contain glacial acetic acid. Ano sagot nyo?
C, sir.
Which are true about nuclear fixative? The correct answer is what? Contain glacial acetic acid. Sabi nga natin sa glacial acetic acid kanina, it precipitates or fix the nuclear part of the cells. And then the pH should be 4.6. The answer here is 1 and 2. Kapag greater than 4.6 and must never contain glacial acetic acid, then this is not nuclear fixative. This is what? A cytoplasmic fixatives. Ito, which of the following is our histochemical stains fixative? Ano sagot nyo?
A, sir.
Yes, A. Except for 10% neutral buffer. Ito, this added as a preservative to formaldehyde. This will prevent its decomposition to formic acid or precipitation to paraformaldehyde. Ano sagot nyo? Ano sagot nyo dito? Letter C, methanol. Ito, formalin is usually buffered at a pH 7 with blank buffer. Anong buffer?
C, sir.
Phosphate buffer. Ano ba yung component ni neutral buffer? Di ba mayroon siyang phosphate? Ito, in tissue containing blood, unbuffered formalin leads to formation of dark brown artifact pigments, pigment granules. These pigments may be removed with picric acid. Number 36. All of the following are metallic fixative except Carnoy's fixative. Alcoholic fixative siya, and it is the fastest fixative. It is used for the preservation of what? Glycogen and microincineration technique, and it is also used to fix sputum since what? It coagulates mucus. Sagot nyo? Genres fluid or alcoholic formalin. Ito naman, excellent for bone marrow, hematopoiesis, and intercalated discs for cardiac muscles, and microanatomic fixative for pituitary gland and blood-containing organs such as spleen and liver. Correct answer is Zenker's-Fulmore. Si Hayden-Hines Sousa solution, a perfect fixative for small biopsy, skin biopsy. They are perfect fixative for skin biopsy. Ito, recommended for demonstration of chromatin or Golgi bodies, mitochondria, mitotic figures, RBC, and colloid-containing tissues, or GMRC. It fixes GMRC. Ano sagot niyo dito?
A, sir.
Records. Yes, correct. Or Muller's fluid. Si ORTS naman is used for degenerative study of disease and for rickettsial infection or a tissue that is infected with rickettsia. ORTS fluid. All are true for Bowen solution except what? Ano sagot niyo dito?
D, sir.
D, yes. Sikat na sikat, no? It does not fix kidney structures, lipid, and mucus. But it is a yellow stain useful when handling fragmentary biopsy. Bowen solution preferred fixative for tissues to be stained by Mason's trichome for collagen, elastic or connective tissue. It preserved glycogen, but does not fix kidney structures, lipid, and mucus. Ito, which of the following is correct procedure of freeze drying? Ano sagot niyo dito?
A, sir.
Letter A. Now, freeze drying and freeze substitute, they are the physical type of fixation. Kapag freeze drying, i-abrupt mo siya, i-subject mo yung tissue or abruptly freeze them or quenching at negative 160 to negative 180. Again, negative 160 to negative 180. And eventually, i-remove mo abruptly din yung na-form na water crystals niya by subjecting it to a higher temperature, which is negative 40. By what? By vacuum pressure? By a vacuum. Sublimation by a vacuum at negative 40 degree. Kapag freeze substitution naman, same lang siya sa freeze drying except for the secondary step, for the second step, which is sublimation. Instead of subjecting it to vacuum or put it into vacuum, gumagamit siya ng Rossman's fluid. Pero the quenching is the same. Negative 160 pa rin, 160 degree. 42, considered to be the most rapid fixative and it fixes the hydrates at the same time. Ano sagot niya dito?
B, sir.
Carnoise. Tandaan niya lang carnoise fluid. It fixes and hydrates tissue and it is considered to be the most rapid fixative. Ito, primary fixatives for electron microscopy are what? Lahat to. Letter D. Tandaan, glutaraldehyde, osmium tetroxide, and paraformaldehyde, they are fixative of choice for electron microscopy. All of these. Ito, what organ should not be dissected before being fixed? Ano sagot niyo dito? Eyes. Why? Because of vitreous humor. Kapag napop yung vitreous humor, then nawawala yung architecture ng eye. And so with brain, dapat i-fix mo na before ita-dissect. And ang fixation ng brain, fixation time of brain is sobrang tagal. And it should be fixed before dissected. Eyes and brain. Ito, for fixation, tissue should not be more than 5 mm thick except in blank. In which cases tissue slices may be 1 to 2 cm thick with a minimum squeezing and handling. Usually, kapag nag-slice tayo ng tissue for fixation, less than 4 mm or 5 mm. Pero may exemption. In what cases? Ano sagot nyo?
Lung edema, sir.
Lung edema. Correct. Ito, decalcification should be done after blank and before blank to ensure and facilitate normal cutting of section. Ano sagot nyo?
C, sir.
Yes, letter C. After fixation and impregnation. Sorry. After fixation and impregnation. Letter C. Mali ata yung nabigay kong ganun. Answer key. 46. 47. Hindi, sakto lang. Fixation and impregnation. It decalcifies and softens tissue at the same time. Ano sagot nyo?
Perennis.
Parenis. Si Von Ebner, ano ang unique sa kanya? Unique si Von Ebner because of hydrochloric acid and what it uses in a surface decalcification. it decalcifies a tissue block sa surface lang ng tissue blocks. Si von Ebner, meron siyang hydrochloric acid. Ito, simple, reliable, and convenient method for a routine purposes to detect the presence of calcium in decalcifying solution. A, physical test. Ano sagot nyo?
B.
Radiologic.
B, radiologic test. C, chemical test. And D, ion exchange ion exchange resin. Si radiological test kasi, it is the ideal form the ideal method in order to detect the presence of calcium. It's the ideal siya. Perfect talaga siya. But it is expensive. Ang simple but reliable, convenient method for routine purposes to detect the presence of calcium is actually chemical test. Again, chemical test. Si physical test kasi, nagkakaroon siya ng false microfracture kapag kuha ka ng tawag doon, tutusukin mo yung tissue, tapos kapag malambot yung tissue, complete decalcified na tong tissue na to. Hindi, makikita yun sa microscope na especially kapag yung tissue is bone, and makikita yun sa microscope na yung tinusok mo as a microfracture. Baka ma-misdiagnose siya as baka may osteoporosis yung patient. So, it is not reliable. Si chemical test, reliable siya, simple, reliable, and also convenient. Why? And it's not expensive kasi it uses only two reagents. First is the sodium hydroxide and saturated aqueous ammonium oxalate. Ngayon, paano to? Kukuha kayo ng 5 ml of the discarded use decalcifying agent, and then kuha kayo ng litmus paper. Since ang decalcifying agent is acidic in nature, makikita mo dun na yung litmus paper will turn into red. Ngayon, drop by drop, you have to neutralize it by adding sodium hydroxide drop by drop. Kapag nakita mo nagte-turn ng blue yung litmus paper, and then upon turning blue, tas cloudy yung solution or medyo turbid, that signifies that you need to re-decalcify the tissue. Hindi pa kompleto yung decalcification ng tissue. But kapag clear naman, that doesn't mean na complete yung decalcifying niya. Pag-add ka ng saturated aqueous solution, saturated aqueous ammonium oxalate, and mag-add ka ng 0.5 ml dun sa clear na discarded na fluid. And then kapag 30 minutes at clear pa rin, ibig sabihin, mag-proceed ka na sa ibang steps or complete na yung decalcification niya. Pero kapag cloudy pa rin, then you need to re-decalcify the tissue again. Yun kapag chemical test. Only needs two reagent, the ammonium hydroxide and the saturated aqueous ammonium oxalate. Again, simple, reliable, convenient method for routine purposes to detect the presence of calcium in the decalcifying solution. It's chemical test, not radiologic test. Now, all of the following are solutions used for electrolytic decalcification except what? Ano sagot nyo dito? Ano sagot nyo dito? Except citric acid. Sa electrolytic decalcification, it uses 88% formic acid, concentrated hydrochloric acid, and distilled water, but not citric acid. Ito, a blank blue discoloration of copper sulfate crystals will indicate full saturation of the hydrating fluid solution and should be changed with fresh solution. Nabanggit na pala yung sagot. Kapag may blue discoloration na of a copper sulfate crystal, then it means na saturated na yung dehydrating fluid, and it requires another fresh solution. That's blue. Recommended for a routine dehydration of tissues, it is considered the best dehydrating agent because it is fast acting, it mixes with organic solvents, and it penetrates tissue easily. Ano sagot nyo? Letter C, ethyl alcohol. Sa isopropyl alcohol, sobrang versatile niya. It is a substitute of ethyl alcohol. Si butyl alcohol naman, it used in plants. Si methyl, toxic siya, or it can cause blindness. Toxic alcohol dehydrating substance. Ito, it dehydrates and cleans tissue at the same time. Ano sagot nyo? Both of these. Again, letter C to. It dehydrates tissue at the same time. Letter C. Letter C, both of these, both dioxin and tetrahydrofuran. 53. It is the most commonly used and most rapid clearing agent in histopathology section. Letter A, xylene. Less than what? Less than 15 to 30 minutes. Excessive exposure to this clearing agent may be toxic and may become carcinogenic. It damages the bone marrow and cause aplastic anemia. Letter B, benzene. Ito, this clearing agent is recommended for tough tissues, nervous tissues, lymph nodes, and embryos because it causes minimum shrinkage and hardening of tissue. Ano sagot nyo? Ha?
Chloroform.
Yes, correct, chloroform. Recommended for the CNS and cytological studies, particularly of smooth muscle and skin. It becomes milky upon prolonged storage and should be filtered before use.
Cedarwood oil.
Good, cedarwood oil. The impregnating medium should be at least blank the volume of the tissue. Ano sagot nyo? It should be what? It should be 25, the volume of the tissue. Yes. In manual paraffin impregnation embedding, what? A, impregnation. Sa impregnation, 2 changes of wax at 15 minutes interval. Complete embedding approximately 2 hours. B, sa impregnation, 2 changes of wax at 10 minutes interval. Complete embedding approximately 2 hours. C, impregnation, 4 changes of wax at 15 minutes interval. Complete embedding approximately 3 hours. D, impregnation, 4 changes of wax at 10 minutes interval. Completely embedding approximately 2 hours. Ano sagot nyo?
C.
Yes, C. Wow, C. Okay. The paraffin oven must be maintained at temperature blank, the melting point of paraffin use. Ano sagot nyo? Yes, A. 2 to 5 degree centigrade above. Wax that has trimmed away from the impregnated tissue may be melted by heating at 10 degree to 105 degree. Pero ang tinatanong, may be melted and blank for the future use. Ano, kasunod dun sa melting? A, filtered with coarse paper. Filter paper example is the green number 904. B, heated at 100 degree to 105. C, cooled. And D, none of these. Yes, letter A. It should be filtered. Then, ilan nyo lang gagamitin ang tissue, ang wax. Ilan pwedeng gamitin ang wax? Twice lang. Kapag twice na, hindi na pwede. So the correct answer here is filtered with coarse filter paper, green number 904. Ito, a substitute of paraffin, which is a mixture of a highly purified and synthetic plastic polymer. Ano sagot nyo dito? A, para plus. B, embedded.
Para plus.
Yes, correct. Para plus. Ito, substitute of paraffin. Take note nyo yung melting point niya. Si para plus has a melting point of 56 to 57. Mixture of highly purified and synthetic polymer. Si embedol, melting point of 56 to 57 degree. Dalawa siya. Either bioloid, which is used for the eyes, and si tissue mat, a product of paraffin, ang unique sa kanya is containing rubber. Kapag nakita na kayong sa tanong as a it contains a rubber, then tissue mat. Kapag ester wax naman, with a melting point of 46 to 48, it is insoluble in 95% ethanol, hence can be used for impregnation without prior clearing of the tissue. Diba, kapag i-skip mo ang clearing, then the appropriate paraffin to be used is the ester wax. Kapag may without prior clearing of the tissue. Ito, water-soluble wax naman, ang melting point niya is 38 to 42 degree or 45 to 56. The common example here is the carbo wax, which most commonly use water-soluble wax suitable for enzyme histochemical studies. Pero kapag carbo wax, wag ka nang gagamit ng ng flotation na tubig. Hindi mo na siya i-float kasi soluble siya sa water. What is the preferred method for a whole section of the ice? Is it A, dry celloidine, B, wet celloidine, C, both of these, and D, none of these?
A.
Try.
Dry celloidine. Huwag kayong malilito sa bioloid, si ice lang siya if kapag yung tinanong paraffin, substitute of paraffin. Pero kapag celloidine naman, then dry celloidine is for the whole section of the eye. Ito, the wet celloidine method is recommended for all the following except what? Eyes, of course. Ito, most commonly used water-soluble wax suitable for enzyme histochemical studies and cytologic details are excellently preserved. Anong sagot niyo dito?
Carbo wax.
Carbo wax. Carbo wax.
Yes, carbo wax. A method of embedding that is really used except when dehydration is to be avoided. Sabi kanina dun sa substitute of paraffin, yung ester wax it can be used if the clearing steps is avoided. Pero ito, dehydration is to be avoided. Ano sagot niya? Ano sagot niyo?
Paraffin.
Ang sagot is gelatin. Gelatin ang sagot. Again, kapag gelatin ang sagot. Kapag dehydration is to be avoided. Number 66. In a double embedding method tissue is what? A,
B.
letter B. Huwag yung pagbalik except to infiltrated with celloidine and then embedded with paraffin. Kasi yung classmate ko na pagbaliktad niya, ESIP. Embedded with celloidine and infiltrated with paraffin. Except to infiltrated with celloidine and then embedded with paraffin. Letter B. Ito. An embedding mold that consists of two L-shaped strips with heavy brush, which the size can be adjusted. Ano sagot niyo dito?
Yolk hearts.
A.
A. During embedding, this is important for demonstration of proper morphology.
D.
Or D. Ito, when cleaning tissue blocks, they must be surrounded with what?
B.
B, 2 millimeter. For our routine works, paraffin sections are usually cut between blank in the thickness for histologic procedure.
B.
Yes, correct. Sana all. After cutting sections are floated out on a water bath set at a temperature of 45 to 50 degree, approximately blank, then the melting point of the wax is used. Ano sagot niyo?
B. B.
It's 6 degree to 10 degree lower. Kasi ganun na nga eh. Kapag higher yan, mamimelt yung paraffin. So, dapat lower. Again, 6 to 10 lower. It is most commonly used adhesive for paraffin section. Ano sagot na dyan?
B.
Letter D, Mayer's egg albumin. So, what is added to Mayer's egg albumin to prevent the growth of molds?
Thymol. D.
Thymol, D. So, most widely used as a section adhesive for immunohistochemistry. Ano sagot niyo? Letter B, polyl lysine.
Poly-L.
Huwag niyong pabalit na rin si APES sa polyl lysine. Coated slides are very useful in cytology, particularly cytospin preparation of proteinaceous or bloody material. A, polyl lysine, B, starch base, B, Mayer's egg albumin, D, APES.
APES.
APES. Ano nga ang meaning ng APES? Anyone? 3-amino-ethyl ethoxycycline. 3-amino-ethyl triethoxycycline. Causes triethoxycycline. Causes of brittle or hard tissue. 1, prolonged fixation. 2, prolonged dehydration and tearing. 3, prolonged paraffin infiltration. C, overheated paraffin oven. 5, tearing out of tissue before actual fixation. Ano sagot nyo? C, yes. C, all of the above. Curing agent turns milky as soon as tissue is placed in it. Due to what?
A.
A.
A, water not completely removed due to incomplete dehydration. Ito, a process of giving color to a section using aqueous alcoholic dye solutions.
A.
Nice, A, direct staining. Ito, a process whereby the action of dye is intensified by adding another agent or a mordant. May mordant na siya.
B.
B.
Yes, indirect staining. A process whereby the tissue is first overstained and that excess tissue and the excess stain is removed or decolorized from unwanted parts of the tissue until the desired intensity of color is obtained.
Regressive.
Boom, regressive. Huwag niyong pagpaliktarin ang regressive at progressive. Regressive. Ito, all of the above. Intravital staining of a living cells is done by injecting the dye into a part of the animal body, producing a specific coloration of the cell, particularly those of reticular endothelial system. Common dyes used for this purpose are lithium, carmine, India ink. All of the above. All of the above, did answer nyo? Yes, letter D. Cytoplasmic stain, except as common cytoplasmic stain. Ano sagot na dito? What?
A.
Yeah, except for neutral red. It is considered to be the best vital stain.
Neutral red.
Neutral red. Yeah, sakto. Wow, vital dye that is used in demonstrating mitochondria.
Janus green.
Janus green. It is the active dye coloring agent in hematoxylin solution. Hematin. Please lang. Hematin to E-I-N, hindi hematin. Letter A. Ito. All fixative can be used for HME staining except OA. Sino dito ang osmic type of fixation? Flemings. Pag osmic type of fixative, don't use it if the tissue is for H&E staining. Ito, all histologic dye derived from an extract from the female bug Dactylopus coccus costa o Coccus cacti.
A.
Letter A. Si hematoxylin is hematoxylin campecyanum. Si orsin naman derived from a certain a bacterial vegetable extract or lichens. Yeah, or si orsin derived from vegetable extract. Si saffron naman Coccus savitus. Again, si coccineal dyes is other term for Karman dyes. Same lang sila. Karman dyes or coccineal dyes. Ito. Acid alcohol used in a routine H&E staining act as what? Si acid alcohol daw act as A. mordant, B. staining, C. bluing, D. differentiator. Ano sagot nyo? Yes, correct. Differentiator. All of the following are artificial ripening agent for hematoxylin except molybdenum. Si molybdenum ba ano siya? Mordant siya. Hindi siya artificial ripening. Again, molybdenum is a type of mordant. Hindi siya artificial ripening. Letter D. In hematoxylin eosin, H&E stains, these stains act as the acid dye. Eosin. It acts as the acid dye that binds to basic part or to the acid part of cells. Ano siya? Eosin is the acid dye that binds to the basic part of the cell, which is the cytoplasm. Now, in hematoxylin naman is a basic dye that binds or act in the acid part of the cell, which is the nucleus part or nucleic acid. Ito. A bluing is a process of shifting the color from reddish to purplish blue by the application of a weak alkaline solution. Bluing is achieved by immersion in all of these. Again, bluing, ammonia water, lithium carbonate, Scott's tip, water substitute. Ito, aluminum hematoxylin is recommended for progressive staining of tissue, but they can also be used for regressive staining. All of the following are alum hematoxylin except C. Saan kabilang si Wigertz and si Hayden Haynes? Kung hindi si alum, iron, correct. It is the oldest stain of stains that stains amyloid, cellulose, starch, carotenes, and glycogens.
Iodine.
Please, wag lang yung oldest stain. Sabihin niyo, oldest stains, then stains amyloid, cellulose, starch, carotenes, and glycogens. That's iodine. Most common method used for the demonstration of basement membrane, particularly the glomerular basement membrane. Ano sagot nyo?
A.
Yes, pass. Acid periodic shift. It is used in a Cajun's method of staining amyloid and may be utilized as stains for axis cylinders in embryo. Axis cylinders. Congruent. The most sensitive lipid stain known.
Sudan black.
Yeah, Sudan black. Recommended in dermatological studies demonstrated the fineness and most delicate fibers in skin. Excellent stain for elastic fiber. or same. What staining used for the demonstration of neuralgia in the frozen section?
Victoria.
Yeah, correct. The Fuel-Jens reaction is the most reliable and specific histochemical stain for
DNA.
DNA. In the methyl green pyronine staining, DNA is stained.
C.
Ready na. The following are the methods for spirochetes staining except
B.
B.
B. Bakit? Para saan yung B? Yung 4. Si white fight. Para saan siya? Leptospira. Ito. Iron-free pigment of hemoglobin found in areas where there is a poor oxygenation participating in the formation of biopigments, that's hemosiderin. To avoid distortion of image, the refractive index of a mountain should be as near as possible to that of the glass, which is what? Ano refractive index ng glass? Ng glass? Letter D. Sometimes they put 1.52. Same lang din yung sa 1.15. 1.18. 1.518. Sometimes 1.52. Pero 1.518. Correct lang din yan. All of the following are resonance mountain media except
Glycerin.
glycerin jelly. Saan siya kabilang?
Aqueous.
Aqueous. Then, pamemorasyon din yung individual na ganun. Na refractive index nila. Kung kaya pa. Sugar is a component of mountain media that is used to what?
B.
Kala ko kayo letter B. It increases the refractive index. Letter B. The cold knife procedure. The cold knife procedure is the most commonly used freezing agents.
A.
Letter C, carbon dioxide. Kapag a liquid nitrogen is the most fastest freezing agent. Pero kapag carbon dioxide in a cold knife, it is the most commonly used freezing agent. Pero the most rapid freezing agent is the liquid nitrogen. Pero the most commonly used is the carbon dioxide. It is considered to be a staining method of school choice for exfoliative cytology.
A.
Who's the father of Papa Nicolau? Father of Papa Nicolau.
A.
Walang father of Papa Nicolau.
George. Charot.
Hindi. Father of exfoliative cytology yun. Hindi siya father of Papa Nicolau method. Kaya kapag who's the father of Papa Nicolau, wala. Pero pag who's the father of exfoliative cytology, din yun. Ang apelyido niya is Papa Nicolau. Hematoxylin is often used to stain what?
Nuclei.
Why? Nuclei is acid part of the cells, and hematoxylin is a basic stains. Si eosin, cytoplasm, is the basic part of the cell, and eosin is the acidic stains. So, in the modified PAP stain, this dye was omitted from the EA formula.
B.
Yes, Bismarck Brown. Huwag niyong kalimutan to, Bismarck Brown. Ito, cytological collection used for a patient with hysterectomy.
A.
Yeah, vaginal scape. Paano kapag nimot ko na din to Paano kapag hormonal evaluation?
B.
Yeah. Lateral vaginal scape. Basis for early detection for early pregnancy, formation of salt crystals in hydrochloric sodium chloride concentration in cervical mucus under the influence of what? Of estrogen. Basis for early detection for early pregnancy. One finding, two palm leaf pattern, dearborization for corlocyte. Anong sagot niyo dito?
B. Charot.
The correct answer here, correct yung B. Pero ito, yung ferning, palm leaf pattern, arborization, pare-parehas lang yan, iba't iba lang yung term niya. Again, ferning, palm leaf pattern, and arborization. Si corlocyte hindi. Nakita yan kapag meron kang warts. May warts yung vagina or the genitals. Corlocytes yun. Papilloma virus pala, corlocytes. Kapag basis for early detection for early pregnancy, ferning, palm leaf pattern, and arborization. They are all the same. No. This autopsy technique is characterized by in situ dissection. Si Rotansky. Ano na nga kay Virchow? Hello? Si Latoul and Masses, si Gon is N block or the organ block, si Virchow, removal of organs one by one. Ito, introduce the empty practice after World War II. Though? 26 medical laboratory for the 6 US Army. Wag 6 medical laboratory tas 26 US Army, baka mapagbaliktad. The Philippine Medical Technology Act, RA 557, was approved on June 21, 1969. It amends Section 16, 21, and 22 of the Republic Act 5527, which was approved on August 31, 1970. Correct nyo ba to? RA 6131. Yeah. It amends the section of RA 5527 it amends the section 16, 21, 22 on August 31, 1970. Si ganun naman sa PD naman, the Presidential Decree 498, which was promulgated or which was signed on June 28, 1974, inamend niya yung section ng RA 5527, section 2, 3, 4, 7, 8, 11, 13, 16, 17, 21, and 29. Again, 2, 3, 4, 7, 8, 11, 13, 16, 17, 21, and 29. Si PD 1534 or the Presidential Decree 1534, inamend niya noong June 11, 1978 ang what? Section 3, 8, and 13 of RA 5527. Ito, the chairman and the members of the Board of Medical Technology shall hold office for blank year after appointments or until their successors shall have been appointed and duly qualified after 3 years. After 3 years. Ito, no person shall practice or offer to practice medical technology as defined in RA 5527 without having obtained a valid what's COR? COR from the Board of Certificate of Registration, from the Board of Medical Technology, provided that registration shall not be required of the following. 1. A duly registered physician. 2. Medical technologists from other countries called for consultation or as visiting or exchange professors to college or universities. 3. Medical technologists in the service of the United States Armed Forces stationed in the Philippines rendering services as such for members of the said forces only. Hindi na daw kailangan ma-issuean ng valid certificate of registration. Sino-sino sila? 1, 2, and 3. Physicians, tapos yung consultation, medical technologists from other country, na consultant siya dito. Then 3, the medical technologists in the Armed Forces of the Philippines, provided na sa grupo lang nila or for the side forces only. Ito, it provides laboratories for accreditation as training laboratories for medical technology students or postgraduate trainees in conjunction with the Board of Medical Technology. Ano sagot niyo dito?
B.
B. Department of Health Bureau o BRL. Mali ata yung na-bold ko dun sa answer key. Pero okay lang. B. Department of Health through Bureau of Research Laboratory. Pero kapag CHED is the Medical Technology Schools. Again, CHED pag Medical Technology Schools, kapag MT training, MedTech training, Department of Health through BRL. Ito, the rating and examination, in order to pass the examination, one must obtain a general average of at least 75%, no rating below 550% in any of the major subjects, not failed in at least 60% of the subject computed according to their relative weights. The answer is letter A. Now, kapag 70 above, sabihin natin 76 ang average mo, pero nabagsak, pero na-feel ko ang HEMA, CC, and Micro, pero 76 ang average ko. Pasado kaya ako? Hindi.
No.
No. Pero kapag na-feel ko ang ganun, kapag na-feel ko lang ang histopath, tapos dalawang major, pass or fail? Pass. Basta 76 above yung nun ko. Kasi hindi naman 60, 20 plus 20, pala 50% at least 60% kasi computed according to the relative weights. 50% lang, hindi siya lumampas ng 60%. Ito, the composition of medical technology boards, ano siya? A, pathologist, medical technologist, medical technician, B, T, pathologist, C, microbiologist, D, pathologist, and D, pathologist and 2 medical technologists. Ano sagot nyo? Letter D. The penalty of suspension of the certificate of registration may be imposed by the board must vote either of these and yeah, majority of vote kapag revocation, unanimous. Ito. Laboratory examination done in a primary laboratory except, ano sagot nyo?
Majority.
Yeah, letter B. Except routine chemistry and gram stain. Kasi secondary na sila. Ito, renewal of the license to operate for non-hospital-based clinical laboratories. Ano siya? Kailan siya non-hospital to ha? Non-hospital-based clinical laboratories. Renewal of license to operate. First day of October to last day of 1st of November. First day of October to last day of November. The last day of November. First day of October to last day of November. Letter A. 124. Any registered medical technologist who shall refuse or fail after due warning by the board to display his certificate of registration in the place where he works, he is violating what?
B. Section
Section 29. Hindi naman nasusunod. Foreign reciprocity is under what section of RA 5727, RA 5527?
27. Section 27.
Section 57. The Blood Banking Law of 1956.
RA
C.
Wow. Yes. An RMT who recently passed the board exam was invited to participate in a medical mission provided by a mobile laboratory. Is he allowed to work given the fact that he did not yet take his oath? A, yes, as long as under the supervision of the physician. B, yes, if she have gained the permission from the Secretary of Health. Yes, because he passed the board exam. No, because he did not yet taken his oath.
D.
D.
B ba sinabi sa inyo?
D. D.
No, sir.
Letter D, no, because he did not yet taken his oath. Kasi hindi ka talaga mabibigyan ng license kapag wala kang proof na nag-take oath ka. Kasi baka by September magte-take oath kayo, then baka virtual din. Tapos kailangan mo picturean yun. Kapag wala kang picture, tapos wala kang proof, hindi ka bibigyan ng ganun, ng license. And para mag-work ka as a medical technologist na legit, dapat may license ka. At hindi ka mabibigyan ng license kapag wala kang proof na nag-oath ka. Kaya during your oath taking, mag-picture kayo, tapos pakita nyo sa PRC. 127, an RMT who recently passed the board exam was invited to participate in a medical mission provided by a mobile laboratory. Number 28, no compulsory HIV testing shall be allowed, but only in the following instances. A, when a person is charged with any of the crimes punishable under the Article 264 and 266 as amended by Republic Act number 8353, 335, and 338 of Republic Act number 3815, otherwise known as the revised penalty code under the Republic Act number 7659. B, when the determination of HIV status is necessary to resolve the relevant issue under the executive number 309. C, when complying with the provisions of Republic 7170 or Organ Donation Act.
D.
Letter D.
D.
Lahat to, no compulsory testing shall be allowed, but only in the following instances. Tawag dun, you should be tested kapag na-meet mo yung criteria ng tatlong to. Pero if not, kapag wala dito, hindi compulsory ang pag-HIV testing. Kaya letter D. Ito, newborn screening program in the Philippines currently included screenings. Newborn screening program in the Philippines currently included in the screenings. Dati-dati, I think 2012, 5 lang to eh. Kaso kasi, ang yung nadagdag is the most common deficiency sa Filipino, sa mga batang Pinoy. Kaya dinagdag ng DOH. Pero dati-dati 5 lang to. Pero ano sa ngayon? Currently included screenings? Ano sagot nyo?
D.
Letter D. 1, 2, 3, 4, 5, 6. Ang nadagdag dito is yung maple syrup urine disease. Ayun, MSUD. Ito ang nagpadagdag. Ito yung memo, DOH memo. MSUD appears to be the most common inborn error of metabolism in the Philippines. DOH memo that includes MSUD in newborn screening panel of disorder. Siya yung nag-include ng MSUD. Letter A. Because of DOH memo 2012-0154 na include si MSUD as a newborn screening program in the Philippines. So currently, silang 6. Pero originally, 5 lang. Kasi the most sa whole world, the most common inborn error in the world is the glucose 6-phosphate. Yeah, the 6PD deficiency. Ngayon, pero sa Philippines, MSUD. Kaya in-include yun. Ito, National Reference Laboratory for Dengue, Influenza, Tuberculosis, and other mycobacteria is RITM. Research Institute of Tropical Medicine. Ito, it refers to the individual from whom a specimen from a drug testing is collected. Anong pangalan niya? Anong tawag sa kanya? Donor or client. According to accreditation of individual partnership, corporation, or association to operate a drug testing laboratory and rehabilitation facility after a compliance with the standard is obtained from what?
Ideya.
Department of Health. Department of Health. Well, PIDEA ba sinabi sa inyo? Or ganun nyo? Department of Health. Accreditation of Partnership Corporation of Drug Testing Laboratory and Rehabilitation Facility after compliance with standards of Department of Health. Again, this is laboratory. Validity of drug testing laboratory. Anong sagot nyo?
A.
Yes, A. Confirmatory lab is 2 years. Screening lab is 1 year. Mandatory drug test except what? As students of secondary. Letter D, correct. Ito. In what phase of a management process does the manager gather together the necessary resources and people and develop an organizational structure to put the formal plan into action? The correct answer here is organizing. Now, in a management process, there are 4 steps. First is planning. After planning, organizing, directing, and controlling. Yung planning, ang ginagawa sa planning, tinatawag siya as the thinking phase of a manager, where the manager evaluates the current situation, the manager or the leader set a goal or objective. And then sa organizing, this is put the formal plan into action. Kapag directing naman, dito na pumapasok yung leadership style mo, or you delegate the work to your employees. You delegate the work to employees. And then controlling yung feedback or your modifications mo kapag meron kang imo-modify. That's planning, organizing, directing, and controlling. Plus, avoid doing harm or minimize harm as much as possible if it is unavoidable. Ano sagot? Ano sinagot niya dito? Non-maleficence. Kapag autonomy, you respect this 4 is actually the principle of ethics. Huwag niyo kakalimutan. Kapag autonomy, you respect the decisions of a person that is capable of making a decision. Kapag beneficence, you do good that is beneficiable to the other. Kapag non-beneficence, you do no harm, or you minimize harm if possible, if unavoidable. Kapag justice, something to do with equity and the distribution of risks and benefits sa tao. So these are the principles of ethics. Now, he created the melody and music of Pamit Him. Sino siya?
C.
Letter A. Sino sa lyrics?
A.
Yes. The Philippine Congress enacted the Toxic Substances, Hazardous Waste, and Nuclear Waste Control Act of 1990. The act seeks to protect the public health and environment from the unreasonable risks posed by these substances. This legislation is under the jurisdiction of what?
Hector.
B. DNR, and the Philippine Nuclear Research Institute. The PRC resolution that reduce the required CPD units, create the required CPD credits units to a minimum which shall not be covered by the CPD requirements.
B.
Dati-rati, 45. Ngayon, letter A, 15 na lang. 17th Pamit National President?
A.
Yeah.
A.
It is a principle of ethics where no person will issue a report orally or in writing without the actual signature of the pathologist or his associates to the requesting physician, except in emergency cases when the result may be released as authorized by the pathologist. Ano yung nalalabag mo sa ethics, sa code of ethics natin?
C.
B.
Oh. A. You have to treat informations are required in a course of my work as strictly confidential. It is still confidential kahit pa may request ay may sign ang pathologies, mas lalo na kapag wala pang sign ang pathologies. Kapag restrict my previous criticisms, views, and opinions within constructive limits, ito na yung mga there's something to do about your workmates. Share my knowledge, expertise in my colleagues, that uphold the law and shall not participate in illegal work. Letter A, that information I acquired in the course of my work are strictly confidential. The answer here is A. So, a type of leadership style that present rules, orders, or other defined instruction to the individual, offers concise and detailed instructions on how to complete a task. Ano sagot niyo dito? Ano sinagot niyo dito?
B.
B.
Yeah, directive siya. When we say supportive, the leader, you're a type of leader that you provide the personal needs or the physical needs of the employee. And when we say directive, you are a type of leader that you present rules and then orders, sabi nga dito, or other defined instructions to the individual. You offer concise and detailed instructions on how to complete a task. That's a kind of leader. Kapag coaching ka naman, coaching leader ka, if you are both supportive and a directive leader, then ang tawag sa'yo is a coaching leader. Kapag neither na both supportive ka o non-supportive ka o non-directive ka, ang tawag na leader sa'yo is delegating leader. Gets nyo? Kapag both supportive and directive, coaching leader ang tawag sa'yo. Kapag non-supportive and non-directive, delegating leader ka. Ito, it maps out the course of an organization for approximately 20 years. It involves tactical and operational plans as well as forming alliances and partnership with key players. Ano sagot nyo? A, short-range planning. B, tactical planning. C, operational planning. D, strategic planning. Ano sinagot nyo?
D.
Letter D. Si operational and tactical planning, they are what? Si short range and ito na ako. Yeah, kapag strategic planning is 20 years. Kapag short range is within 1 to 5 years. Short range and tactical planning, they are the same. Short range or tactical planning. Kapag operational planning ng 1 budget period or within 1 year lang. Again, short range, 1 to 5 years ang plano mo. Then ang same with tactical, since short range or tactical planning. Kapag operational planning naman, lesser than 1 year lang yung plan mo. Kapag strategic planning, then may partnership ka or you plan approximately for the next 20 years. The use of computers and information system to process and communicate information generated in laboratory. The answer is laboratory informatics. Now, kapag informatics lang, ang definition niyan is the science of what? Of processing data through computer. Retrieving it or use the data, that's informatics. Kapag laboratory information system, these are the systems of hardware, software, connections, and communication protocol that handle all information needs of the laboratory. Pag laboratory informatics, information system to process and communicate information generated in a laboratory. Medyo wala silang masyadong pinagkaiba. Pero sundin nyo na lang. Retention of quality control records and requisition as accession logs. Ano yung retention niya? It's 2 years. 2 years. Retention of wet tissue specimens. It's 2 weeks. Ito, a phase of team development that determines standards of behavior and expectations. Ito yung phases ng team building. May mga phases pala ang team building. When we say forming, you have to identify your group mates or your team member. Kapag storming, the team member delegates the task to each individual members. Kapag norming, yun nga, yun na yung expectation mo or standards mo or yung standards of behavior and expectations mo within the member. Kapag performing is you actually the actual when the actual performance or you do the actual work that you have to reach a common goal. Parang yun yung performing. So there are 4 phases of development in team building: the forming, storming, norming, and performing. Norming kapag a phase of team development that determines standards of behavior and expectations, that's norming. That's norming. In hand washing, rinsing hands in downward or upward?
Downward.
It should be downward. Huwag kayong mahilikod dun sa sa mga surgeon, puro upward sila, pero siguro pero sa atin, siguro downward. The correct answer here is downward. Ito, reactivity for, ano sagot nyo dito? Hindi, dun sa reactivity na for. Ano sagot nyo? Unstable if heated, violent chemical change may deteriorate, and shock and heat may deteriorate. The answer here is 4. Yun lang. Thus, before I end this, kamusta pala kayo?
Okay lang, sir.
Ha? Okay na. Before I end this, meron akong gustong share. Baka kasi lalabas din. Sabi kasi ni Vroom's expectancy theory, isa din to sa ganun sa baka kasi tawag dun, additional information na Vroom's expectancy theory. Sabi niya, effort will bring reward. Effort will truly bring reward. And that effort expended will be related to the perceived value of reward. Sa ngayon, nag-effort kayo, and I know 100% na ang perceived value of reward niyan is this. Registered medical technology soon. Yun lang. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.